Tak dengar Diana. Dah boleh rekod dah. Dr. Diana dah keluar Diana ada ke dalam ni? Dr. Diana ada ke? Boleh student confirm kan Dr. Diana ada tidak? Saya tengok tak ada dah.
Tak ada doktor, dia kena remove tadi.
Alamak.
Dr. Diana dah tak ada, Dr. As.
Sebab masa bila saya rekod tadi dia tulis due to technical problem terpaksa remove Dr. Diana. So dia dah terkeluar. Tak apalah, kita rekod dulu. Kita mulakan dululah, sekejap lagi Dr. Diana akan masuk. Sekejap saya cuba contact dia kejap. Okay, saya dah contact Dr. Diana. Since one of us akan pergi untuk vaksin hari ni, Farah Idayu. So I do not want to waste any more time. Farah Idayu dah ready ke?
Yes, doktor, insyaAllah.
Okay, insyaAllah. So I wish you all the best of luck semua student. InsyaAllah sesi akan berjalan dengan lancar. Jangan nervous sangat, jangan takut sangat. Always remember we are here to help you. Kita cuba bagi feedback lah berdasarkan apa yang you all present so that your written report nanti akan jadi lebih baik daripada apa yang ada sekarang. Okay. Alright, so without further ado, I would like to invite Nur Farah Idayu to present to us your FYP project.
Alright, I will share screen.
Okay.
Can you see my slide?
Yes, we can see your slide.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and good afternoon. I bid to all examiner and my fellow friends. My name is Nur Farah Idayu binti Muhammad Asri. I will present about my final year project is a review on morphological and anatomical properties of 4 bamboo species in Malaysia. Let me start my presentation with a little bit background of bamboo. Bamboo is a type of grass belonging to Poaceae family. It's widely distributed in Southeast Asia, Africa and Latin America. There are about 90 genera and 1,200 species of bamboo are found. However, in Malaysia, there are about 70 species found from different genera such as Bambusa, Gigantogloa and Dendrocalamus. There are 2 general types of bamboo which are clumping types and running types. This slide shows the differences between 2 types of bamboo, which is at the left side we have clumping bamboo or known as sympodial and the right side we have running bamboo or called as monopodial. As we can see here, the differences between these type of bamboo, we can spot the different based on the growth of its rhizome. For clumping bamboo, the rhizome develop more radially and a little bit horizontal, while running bamboo grows more horizontally before they are transformed into culm. This is research question that I used to write in my review article, and this will be discuss in my presentation later. So what the objective of my study is to determine the differences of morphological and anatomical properties among 4 bamboo species in Malaysia, which are Bambusa vulgaris, Bambusa blumina, Sargentochlora ligulata, and Dendrocalamus asper. This is important to know because of morphological and anatomical properties of bamboo have been found to huge effect based on their sustainability and the endurance. Other than that, it's also important to gather of all this information because we can determine the best way to use bamboo. Next, I will discuss about bamboo species that I write in my review article. Firstly is Bambusa vulgaris, or in Malaysia we call as buluh minyak. It is clumping bamboo type that native to southern China and Madagascar. It's grown naturally on riverbanks, roadside and wasteland, and it's generally used in light construction such as home, furniture and handicraft. Next bamboo species is Bambusa bulmina, or we call as buluh duri. It's a clumping species bamboo that originates at Malaysia and Indonesia. It grows along riverbanks, hill slopes and freshwater creeks. It also can be used as kitchen utensil, furniture and its edible shoot as a food. Next bamboo species is Dendrocalamus asper, or known as the giant tropical and thick clumping species that native to Southeast Asia. In Malaysia, we call as buluh betung. It planted from low elevation and grow well in variety of soil type. It's used as a building material, as structural timber for heavy construction such as for houses and musical instrument. Lastly is Sargentochlora ligulata. It's a clumping type of bamboo that native to northern part of Peninsular Malaysia, that found in Elaeis, Kedah, Kelantan, and also found at Thailand. It's found in overlook forest, margin of secondary forest and wasteland along the roadside. It used to frame chairs, tables, and screen. Next, I will discuss about morphological properties of bamboo. Morphological properties is important for recognition or identification of all organisms, including plants. As we can see here, morphology properties of bamboo consist of culm section, which are knots, interknots, and culm mold. According to Zakikani, each bamboo species has different morphological properties. I will talk about interknots of culm, interknot is a part between two knots of a bamboo culm. According to Siam, Bambusa vulgaris and Bambusa bulmina share the same interknot length, while the asper and G ligulata also share the same interknot length. The asper and G ligulata have longer interknot, which is 37 cm compared to Bambusa vulgaris and Bambusa blumina. Next, I will talk about thickness of culm mold. The asper, according to Siam too, the asper found as the thickest of culm mold, which is 16 mm compared to other species. This species show that the asper more suitable to make a furniture because the thick of culm mold. Next, I will talk about anatomical properties of bamboo. The anatomical structure was studied because of their relation to endurance, preservative absorption, propagation, and possible microbe, and also colonization pathway. In my review, I just review two parts of anatomical properties, which is vascular bundle. The number of vascular bundles become larger and more widely spaced in the bamboo clump as they progress to middle layer, according to Zakikani. Based on research list, there are 4 types of vascular bundle found in plant. However, G. lingulata has type 3 of vascular bundle since has a single fiber bundle that has been separated, while for Bambusa vulgaris and Bambusa blumina and the Asper have since there has 2 isolated fiber lines on the flower and protoxylem at the ends, and also consists of central vascular strand in sclerenchyma sheath. Lastly, I will talk about fiber strand. From the review, G. ligulata is the longest fiber strand, which is 3,090 and 913 micrometer, followed by D. asper. As conclusion, these 4 bamboos show different morphological and anatomical properties. Generally, the asper has the thickest, longest internode and fiber compared to other bamboo species studied. Therefore, this species is the most suitable to be used as building material, furniture and parquet. My suggestion or recommendation is further research need to be performed on this species as well as for other species to increase the utilization of bamboo in Malaysia, since bamboo is the best replacement for woods and have faster growth rate than trees. This lists some of references that I use in my review article. Then yeah, that's all from me. Thank you.
Thank you, Farah Idayu. Can you show us the first slide which you have the title? Which you show the title? Panel dah boleh ready untuk soalan ya. We have 2 minutes for Q&A session. Right. Okay, Farah Idayu, Dr. Nurizah is a supervisor. Let me have a look over here. Okay, for this review article, do you have main paper or main journal that you have been used as your main reference?
Yes, I do have the main paper.
Okay, which paper?
Siam, wait. I show the references. Zakikani is one of main paper that I refer to.
Alright, so this is the main paper that you are referring to, and then based on this paper, you write a review on different types of bamboo. Is it those different types of bamboo? Is it the same as the one that has been described by Zakikani? Because he mentioned 4 bamboo species. Okay, is it the same species with the one that you have reviewed?
Oh no.
Different.
Yeah, different.
So
Because
yes, continue.
Because based on the paper, Zakikani only write about Bambusa vulgaris,
Okay.
Dendrocalamus asper, and another 2 type of bamboo I can't remember, but the other species I took from another journal.
Okay, so how many additional species that you have apart from the one that have been described by Zakikani?
Pardon, doctor?
How many additional species
Uh,
do you have in your review apart from the one that has been described by Zakikani?
Oh, just 2.
Two more additional species.
Yes.
Do we have any other question from the panel? Dr. Ka, Dr. Diana?
Assalamualaikum, Farah. Can I ask you something?
Salam.
Congratulations for completing your writing and also presentation just now. All right. My question is, why your study just limited to 4 bamboo species? What's the main factor?
Because I just read, I find the related article, but Dr. Noriza asked me to give bamboo species that familiar in Malaysia. So I choose randomly 4.
Randomly. This is not the
Yeah.
top 4.
Yeah,
How
no.
many species that we have in Malaysia actually? How many bamboo species that we have?
According to Muhammad, there are about 70 bamboo species.
17 or 70?
70.
70. 70.
Yeah.
Okay. All right. Now, do you have any idea why you just choose these 4? Is it this top 4 or what?
Yeah, because the other species, I find it's very hard to me to get related topic that I want to review. So I found the most journalists write about the bamboo, the four bamboo species that I choose is easily to get the resource.
Okay. Talking about the ethnobotanical, do you have any idea what is where we can find these 4 species? Means
Macam mana?
instead of state. Okay, 4 species. You mentioned about 4 species now. Okay. Does these 4 species appear or present in each state of Malaysia?
I'm not sure, doctor, but because on my reading state there, wait. For example, for Gigantoplaligulata, we can find at Perlis, Kedah and Kelantan. For other species, I'm not sure, doctor.
Okay, you didn't discuss about the ethnobotanical, right?
Yeah.
Okay, please take into your consideration to add this point in your review article. Maybe we can see why we choose these 4 species to be reviewed in your article, right?
Okay, doctor.
That's all
Thank you,
from me.
Dr. Diana. Thank you,
Alright.
Dr. Diana. Time is not on our side. Masa dah habis. However, Farah Idayu, I would like to remind lah, kind of macam, I want you to bear in mind that you have to justify why you choose only 4. But taking into account that we have about 70 species of bamboo trees in Malaysia kan, tapi you ambil 4 je, you have to justify that probably this is the top most common species or this is the apa-apalah, you have to justify on that. Okay, kita tak
Right.
boleh macam simply only take 4 just because it will be easier for you. Tak boleh macam tu. Okay, you have to do the justification for that. Discuss with the supervisor and see how you can justify in choosing only 4. Alright, thank you, Farah.
Sorry, Dr. Asmida, can I just add one comment there?
Yeah,
Just
sure.
keep on 30 seconds. So I think maybe the 4 bamboo that you chose, is it? I mean, maybe they are important for commercial value. So I think maybe you can justify that if that's the case. Okay, that's all from me. Thank you. Cool.
Alright, thank you, doctor.
Okay, thank you, Farah. Okay, our next presenter is Muhammad Amar. Muhammad Amar Zulhilmi bin Muhammad Saad. Amar, you ready? Kalau ikut senarai saya ni, Muhammad Amar nombor 2, kemudian Muhammad Haris ya.
Yes, doctor.
Amar kuat sikit, tak dengar. We barely hear you.
Doktor.
Dengar tapi jauh je.
Boleh dengar, doktor?
Boleh.
Better than before.
Boleh, tapi kalau boleh kuat lagi.
Maybe your mic.
Angkat suara ke? I
I think you need to be closer to your mouth. I think.
Benda tu dekat sikit dengan mulut.
Dekat sikit your mic. Closer to your mouth.
Try cakap.
You are muted, Amar.
Amar, you're on mute. Amar, the microphone. You can tekan dekat screen. Amar, I cannot unmute for you. You kena mute sendiri. You kena unmute sendiri. Mic rosak. Cuba buka. Okay.
Okay. Next presenter lah, Dr. Asmita.
Muhammad Haris ada?
Ya saya doktor.
Muhammad Haris, boleh you present dulu while Amar is fixing his problem?
Boleh. Boleh
Okay.
dengar jelas ke doktor?
Boleh, jelas. Jelas, thank you.
Okay.
Boleh mula present Amar, Hilmi, Haris? Pening dah saja.
Hello, doktor.
Ya, Amar, we'll make way for Muhammad Haris dulu ya, Amar.
Oh, okay.
Okay. Muhammad Haris.
Doktor, saya ada technical problem sekejap
Technical problem macam tu, Amar okey ke?
Sebab slide saya ni tengah loading. Dia
Lambat.
Back to Amar.
Okay. Dengar tak?
Dengar tapi jauh lah.
Boleh
Kita ada markah untuk vocal quality tau, 2 markah. So if we barely hear you, nanti apa yang you nak sampaikan tu tak sampailah pada panel.
Sebab mic saya macam selalu rosak juga.
Sekarang dah better sikit. Sekarang better.
Now okay? Okay.
Okay, kena tak dekat kat mulut.
Okay.
Okay, you boleh present sekarang, boleh share your slide.
Okay. Ada doktor?
It's coming. Okay dah. Okay, your time starts now. 8 minutes.
Okay. Assalamualaikum and very good afternoon to all lecturers and my fellow friends. Today, I would like to present about my final year project, which the title is Tree Species Composition and Forest Structure at Compartment 15 in Sungai Lalang Forest Reserve, Selangor. My name is Muhammad Amal Zuhimi bin Muhammad Saad and my supervisor Dr. Faizah binti Pardi. So let's start the presentation. For the introduction, I go to the problem statement first. So, there are many anthropogenic factors by the human activity such as logging, mining, and also the social factor like hiking and also picnic that would give disadvantages to the forest ecosystem, which is affect to the species extinction from illegal logging and also affect to the function of the forest and threaten the biodiversity. So, from this study, I gained the knowledge and understanding on the tree species composition and the forest structure. The research also provide the valuable reference for forest assessment or management and improve the skill in identification. There are 2 objectives of the study, which is to determine the tree species composition and the forest and the species diversity at Compartment 15 of Sungai Lalang Forest Reserve. And the second one is to estimate the total of biomass of the tree communities at Compartment 15 of Sungai Lalang Forest Reserve. Okay. From we go to the method. During the pandemic issue, I'm not going to do the quadrat sampling into the tree species. I'm do the data analysis only. So the quadrat sampling into the tree species identification was conducted by the forester. So the first So the first step is the quadrat sampling method, and then we measure the tree of diameter of breast height at 5 cm and above. After that, we collect the sample and brought to the laboratory to identify the species identification. So after that, we just run the data analysis by using the PAS software, which is paleontological statistic and Excel to get the result of taxonomic composition, tree diversity, and also forest structure. Okay, next. This is the study area. The compartment 15 of Sungai Lalang Forest Reserve was located near the Hulu Langat, Selangor. Okay, next. There are 5 important things in the result, which is the first, taxonomic composition. So from this forest, there are 29 families, 49 genus, and 49 species. So Dipterocarpaceae was the family with the most species of 10 species. So the picture on the top right of the slide, we refer to the cesium species with Metaceae that will be the highest individual, 27 of individuals. Okay. So the table below show the comparison of the three forests instead of my study site. Okay. Next, the second is species richness, species diversity, and evenness. So we will follow the range of 1.5 into the 3.5 to get the Shannon index value. And then we also use the range from 0 to 1 to get the evenness of the result. And then there are the one indicates that all species are equally abundant. Okay, that for the forest structure, this is the graph of diameter of breast height, size classes versus the number of individuals. From this graph, it shows the inverse of J-shaped pattern. But however, for the diameter of breast height class 1, which is 33 individuals, is less than the DBH class number 2, which is 99 individual, because of the small tree that dominate to the land. Okay, next. The importance of value index, IVI. So the family and the species of IVI of more than 40 and 10% will be considered as having of absolute dominance. Okay, the table below show the highest of species and the family, which is cesium species, Shorea species 2, Shorea inapendiculata, and for the family is this Dipterocarpaceae, Metaceae, and Abenaceae that contribute to the value of the relative frequency, relative density, relative dominance, and also the importance of value index. Okay. Next, for the biomass estimation, there are 3 values, which is the total of the biomass of 1 hectare compartment 15 Sungai Lalang Forest Reserve, the above ground biomass, and also the below ground or the biomass that will be estimate, which 433.91 time per hectare, 370.30 time per hectare, and 63.61 time per hectare. So the table below show the highest of family and also the species, which is the Dipterocarpaceae, Metaceae, Malvaceae, and for the species,
Mama, you have 1 more minute.
Shorea species 2, Shorea inapendiculata, and also Cesium species. Okay, for the conclusion, the first conclusion can be complied to the first of the objective, and the second conclusion will be complied to the second objective. Okay, next, for the recommendation, I suggest to be increase the larger of sampling area in the future study, and also increase the number of parameter used, such as the soil factor, and also I suggest to improvise the forest management team by monitor the condition of the forest, and also this study will be the potential as the restoration forest. Okay, next, this is my reference that I choose. Okay, that's all for me.
Okay, thank you, Amar. I will open the floor for question and answer session. Do we have any question from the panels?
Okay, Amar, I would like to ask you congratulations again for come to the stage. Okay, however, I need to ask you some question regarding to your data. Please show me your data.
Uh,
The data tu. Tadi. Show me the table that contain your finding.
finding.
Okay.
Which
The
one?
second one. The second one.
Second one.
All right, this one. Okay.
Okay.
Most of the data that I saw here, I see here
Yeah.
now. Okay. Do you have any replication?
Replication.
Means this is only a single reading, or you have more than one reading? How many times you collect the data?
Which is the paper or what?
The data. This
The
is
data.
yes, the data. How many times
This is
you
your
collect?
very own data, right? This is not an extract from of data from other sources.
Because all the data I get from the different of the paper, so I
Different of the paper. So meaning
Yeah.
that you have more than one paper, right?
yeah.
You collect.
I
So
have
meaning
I have around 70 paper. I use. Yeah.
this is the okay. Talking about the replication. So means
Yeah.
that you have more than one replication. Means you have more than one reading, isn't it?
Yeah.
Okay, when talking about replication, so means that now you have replication means in this case I'm not sure how many that you have exactly. So when talking about replication, you need to proceed for the statistical analysis. But here I can't see any standard deviation or standard error presented here. Do you get my idea? Does you conduct any statistical analysis?
Because my data run into the Excel, and
Yeah, I know.
yeah.
But you have the replication. When talking about replication, means that you need to
Yeah.
know how accurate, how precise your data. So to know that thing, you have to perform the statistical analysis. What is the standard error, or at least standard deviations from your data.
Oh.
Okay, show me the next table, please, Amar.
Okay.
Same goes to here. You only have a table, but incompletely analyzed. Okay, you need to have the error bar to show how different one reading to another reading. Then we can put the discussion there whether your finding or your data is accurate or not. So that's the reason you need to have the statistical analysis. Even if this is a secondary data. I know this is a secondary data, means the other person collect the data to you and then you have to analyze. But when you have the data set, so you need to analyze using the statistical analysis. Okay.
I agree with Dr. Diana. We do realize that when students are obtaining data from other sources, especially the journals, you tend to cut and paste the data without analyzing. You do put some effort on your part to analyze the data, because once you analyze the data, you will be having the statistical analysis with it, especially standard error of means. Okay, the list would be standard error of means or error bar here. Kalau tidak, if you don't have error bar in this graph, we cannot see the significance, okay, between one class to another.
I think I think I do the statistic of data because I ran into
Just
the
get me here.
Yeah,
Yeah.
it has to be reflected here. Kena ada bar dekat sini. Error bar has to be here.
in the
The
graph.
graph.
Now, the
Oh.
graph, the table, and so
In the table, table pun all reading should have apa tu orang kata SEM ataupun standard deviation depending on what you are looking at.
Oh, okay.
Betul ke Dr. Diana?
Exactly. Ahmad, can you show me the problem statement? Okay. At here, I would suggest you for your written report, okay, to add at least any statistic to show the seriousness of the issue. You bring the issue here, you bring the issue to discuss or to review, to solve here, but you didn't mention how serious the issue. So please try your best to find out the statistical analysis or the statistical prevalence about the issue. So the point will help you to justify to the reader that your issue, the issue that you bring here is serious issue.
Understand?
And then I would also to suggest you that please include any citation when talking about problem statement. Means that you need to have the citation. Means you refer to someone, okay, to others' work. So citation should appear in your problem statement actually.
Okay.
All right.
Okay, thank you, Dr. Diana. Dr. Ka, do you have any comment?
I just wonder, like this. Okay, Amma, you did mention earlier that you obtained the data from somebody that helped you or assist you in getting the data, right? Or are you taking it from the literature, or or someone at the forest park gave you the data?
I get the data from the Forestry of Malaysia, Department of Malaysia.
I see. Okay, and then regarding replications, I think it is a bit quite difficult for you to conduct, or the ranger there or whoever are responsible for data collection there to obtain the replication. But I think like what Dr. Diana and also Dr. Asmina mentioned just now, I think what the best way that you guys can do this is by conducting the same experiment, maybe 3 times there, and then after that you got the average value and you can obtain the standard error or standard deviation. So that's the best way, I think. So that is my comment regarding if you want to conduct replication, because your study is I think slightly different from others because you have to cover maybe wide coverage area there. Okay.
I think from my 1 hectare of Sungai Lalang Forest Reserve, divided into the 10 of plot.
Oh, I see. Okay.
10 plots of the
so
study.
it is manageable to obtain that. Okay.
Yeah.
So I think the best way is maybe you can try to give some instruction, okay, to the data collector to conduct the data collection maybe in triplicates, for instance, okay, so that you can conduct your statistical analysis. At least you must have the average value plus and the plus minus of the standard error as well as the standard deviation.
Yeah.
All right. Okay. Thank you.
Okay, thank you, Dr. Khairul Asfar. Thank you, Ammar.
Thank you.
Our next presenter is Muhammad Haris Zafran. Haris, are you ready?
Yes, Doctor.
Okay, you may present now. You may share your presentation with us. Okay, we can clearly see your slide. Your time. Yes, we can see your slide. Your time starts now.
Assalamualaikum and good afternoon to the panel of examiners and my final year project colleagues. My name is Muhammad Haris Zafran bin Zainal Abidin, and today I would like to present on my final year project with the title Colchicine Induced Mutation in the Capsicum annuum and its effect on the plant morphology. Mutation induction in plants, or simply known as mutation breeding, is an effective tool for crop improvement. The use of chemical and physical agents is an important approach for inducing the mutation artificially. Therefore, colchicine was used as a chemical mutagen in this study to mutate the capsicum annuum. There is a constant increase in the demand for the capsicum annuum, so the efforts need to accompany to increase the production. One effort that can be done is to improve the quality of fruit by mutation breeding using colchicine. Therefore, the objective of the study are to conduct a comparative analysis on the effects of colchicine treatment towards the growth rate, cytological and morphological of capsicum annuum at various exposure duration. Then we move to a little bit of literature review. A mutation is a heritable alteration in a live organism's genetic makeup. Mutation can be spontaneous or purposely produced with the use of physical or chemical mutagens which function to develop the mutants with desired features. Next, colchicine is the most utilized antimitotic agent in the plants for the activation of polyploidy as well as change the chromosomal structure. Colchicine is a major mutagen that stops or alters the development of microtubules and double the chromosomes number. Then for analysis mutation, there are three types that can be done, which is chromosomal aberrations, mitotic index, and nuclear abnormalities. Chromosomal aberrations are defined as changes in either chromosomal structure or total number of chromosomes, while mitotic index is defined as the total number of dividing cells during the cell cycle. And then for nuclear abnormalities is morphological changes in the interphasic nuclei cause the behavior of the study agent. And then genus Capsicum, also known as a chili or pepper, belongs to the Solanaceae family. Capsicum species are diploids, mostly having 24 chromosomes. Among the numerous well-known capsicum varieties, Capsicum annuum is one of the world's most important agricultural crops. Then we move forward to the methodology of the study. First is the preparation of the capsicum annuum seeds, and next is the preparation of the colchicine solutions based on different concentrations. And then the seeds were soaked in the different types of colchicine concentration at different time exposure. Then the seeds were germinated in the pots and polypropylene bags. Lastly is doing the macroscopy and microscopy observation. Next, this is the result for the seed germination. Based on the results of 24-hour treatment, the control treatment had the highest germination rate and germination percentage, and followed by 0.025% treatment as the second highest. The germination rate and germination percentage started to decline after treated with 0.05% of colchicine, and the lowest reading was seen in 0.1% treatment. Then for 48-hour treatment, the result also same as 24-hour treatment, where control treatment have the highest germination rate and germination percentage, and it started to decline after treated with 0.05% of colchicine, with the lowest reading was seen in 0.1% treatment. Then we move forward to the results of the root length. According to the results, the control treatment of 24 hours exposure period show the highest mean of root length of Capsicum annuum between all groups, including the treatment of exposure period at 48 hours. Based on the previous study on Chandaeng orchid by Cat1 and Tikato, it's revealed that the putative tetraploid plant had a much lower number of root length than diploid plants. Cytological analysis of the root tips revealed that tetraploid plant had 2 times the number of chromosomes than the control. Polyploid plants usually have a bigger and a longer organs than diploid plants, which are seen in a variety of plant species. However, it suggests that the results may not be the same for all plant species, and they may be depending on the kinds of plant tissue. Then this is the result for the plant height. Based on the result of 24 hours treatment and 48-hour treatment, it was discovered that treatment with 0.075% of colchicine concentration at 24 hours exposure period improved the plant height compared to the control. compared to the other treatment and including the control. Therefore, the optimal colchicine concentration for increasing the plant height is 0.075%. Other than that, the results also reveal when extending the exposure period to the colchicine, it will reduce the plant height. Thus, the ideal exposure time is 24 hours or less. And then next, we have the results for the leaf length and width. Based on the results, the best treatment for improving the length and the width of leaf is by using 0.075% of colchicine concentration at 24-hour period exposure. Then for 48-hour time exposure, the treatments of 0.025% and 0.075% exhibited a significant improvement in the leaf length and width compared to the control treatment, although not as much as the 0.075% of 34-hour treatment. In general, the best colchicine concentration to choose for improving the leaf length and width is 0.075% at 24 hours of exposure. Then this is the result for the mitotic index. Based on the results, the treatment with 0.075% of colchicine at 24 hours time exposure resulted in the highest mitotic index value when compared to the treatment of the other groups, including groups of 48-hour treatment, while the lowest mitotic index was shown by 0.1% of colchicine treatment at 48-hour exposure period. As a conclusion, the changes in the mitotic indices of treated roots which either increase or decrease, it's implied that one effect of colchicine treatment is a shift in the balance of the component that govern the mitotic cycle in the root cells. According to the Balog 1982, colchicine has been shown to enhance the frequency of polyploidy cells. Because polyploid cells have more chromosome and hence more genetic material than diploid cells, the mitotic cycle and the mitotic index may be influenced by this increase in the genetic material. Lastly, this is the result for the chromosomal aberrations for 24 hours treatment. The only treatment that demonstrated chromosomal aberration was 0.075%. And then there is a technical issue for the other treatment groups, resulting in no results. For 48-hour treatment, the highest value of chromosomal aberration is 0.5%, and then followed by 0.0225% as the second highest in the groups. And then there is only 2% of chromosomal aberrations value for 0.075% of colchicine treatment.
Haris, you have one more minute.
Next, this is some example, the results for the chromosomal aberration that I have in the study. There is C metaphase, and also lagging chromosomes. And next, there is a clumping anaphase and also a sticky chromosome. And lastly, we have a binucleated cells of Capsicum annuum. For a conclusion and recommendation in this study, the introduction of colchicine at certain concentration and exposure time induced the significant changes in the morphologies, such as seen in the leaf size and the plant height as compared to the control treatment. However, there is also inverse reaction of colchicine as compared to the control, which can be seen in the reduction of germination rate and germination percentage of Capsicum annuum seeds as the colchicine concentration increase. And this is my references, and that's all from me. Thank you.
Thank you, Haris. Can we have a look at your first slide?
Okay.
Okay, in general, based on your results and based on your presentation, what can you say about the effect of colchicine on the plant morphology, the most significant results?
The most significant result, I can give two examples. The first is the plant height. The purpose we study this, we want to increase the plant height, where it will give the benefits to easily to harvest, and then it also will help the plants avoid from the plant pests, and then for the leaf traits, we want also to increase the size of leaf, where or surface of area of leaves, where it will help the plants to get a better site for photosynthesis rate when have a broader surface area, which it can absorb more light from sunlight when have more chloroplast, and then with and then have a larger surface area of leaf will give benefit to the plant, where it will improve the gas exchange as the stomata increase on the surface area of plant.
Okay, thank you Haris. Panjang penerangan dia. Thank you so much. In a nutshell, would you recommend inducing colchicine to the chili plants?
Sorry, Doctor?
So as a conclusion, would you recommend inducing colchicine into the chili plants, into the capsicum annuum plants, based on your results? So
what concentration that I recommend?
No, in general, would you recommend? Tak jelas suara saya. Okay, in general, would you recommend inducing colchicine into the capsicum annuum? Saya tahu dah jawapan dia, cuma saya nak tahu.
Yes, I would like to recommend since the results that I get 0.075% shows the most improvement in the traits of plant morphology, such as increasing in the plant height, increasing in the length and width of leaf, and also to the microscopic observation, which increase to the Mighty index.
Okay, good. Thank you, Haris. Based on your answer and also your presentation, I'm sure that you have put forward the evidence of the benefits of having induced capsicum. Saya pasang timer tu. Induced capsicum to capsicum annuum. Alright. Do we have any more questions from the panels?
Just one question from me, Haris. What is the best or the optimal height of the capsicum annuum plant that would prevent the plant from being infested by, the pest?
I don't sure the best optimum for the plant height, but I would like to suggest for as you can see, the control shows the 1.4 centimeter compared to the 0.075 of 24 hours, which increase into the 2.95 centimeter. So as what I can say, it will give more benefit to the plants if it will be more higher to the other plants. First, it can avoid it from the plant pest, and also it will increase the successful in the competition of lights. But for the best height for improving the plant from the plant pest, I'm not sure how much the height we want to have.
Okay, it's okay. Haris, I think you can try to search it in the literature. I think that must be there. All right, thank you very much. Cool.
Okay, thank you, Dr. Khairul. All right, we'll have our next presenter with us, Nur Akilah binti Muhammad Junaid. Akilah, you ready? Nur Akilah, thank you, Haris. Okay, Akilah, you may share your slides with us now.
you see my slide, Doctor?
Yes, we can. Okay. You may begin. Your time starts now.
Assalamualaikum and good evening to all panels and all my fellow friends. My name is Nur Akila binti Muhammad Junaid. Today I will present about my final year project entitled Factor Affecting Stress Behavior in Cat During Veterinary Visits. My supervisor is Dr. Nordadia Meti Mat Yusuf. The background of my study is about cats, domestic cats, Felis catis, which are one of the most prevalent companion animals in the world. The veterinary care is essential to maintain the welfare of cats. Regular veterinary visit for preventive care and early in disease could enhance pets' quality and lifespan. The problem statement in my study is veterinary visit can be stressful for companion animal, especially cats, because lack of understanding of cat stress behavior can negatively impact their welfare, as proper management to reduce the stress cannot be done. They also make the veterinary team feel pressure when handling cats because they're less cooperative when do the examination. So the significance of my study is to provide an insight on the magnitude of stress behavior and help the veterinarian team to provide the proper management to reduce cat stress while handling them, and help to improve cat welfare and provide the cats with the best experience during the veterinary visit. The first objective of my study is to determine the stress level of cats during veterinary visit based on their behavior in solitary cage at different time points. And the second objective of my study is to determine the effect of cats' individual characteristics on the display of stress behavior in the veterinary clinic. My study site is conducted in a veterinary clinic located in Ipoh, Perak, and 22 unmatured cats aged between 5 months to 5 years old were used in the study. We also use questionnaire that used to obtain the information about cats and were placed at the clinic front desk for the owner to read and fill up once they decide to participate in the study. The first method, we distribute the questionnaire to the cat's owner, and after get the approval from the cat's owner, we start the stress behavior observation, which is cat stress score, approach test, and play test. These three stress behavior observation were conducted within one hour. Then after we conduct the stress behavior, we collect from observation and video recording, and all data were analyzed in SPSS software. This is the sample of the questionnaire that we distributed to the cat's owner, and we obtained the breed, age, color, sex, and the reason for cats visiting the clinic. And the consent form for approval from the owner was also included in the questionnaire. This is the cat stress score that we use in this study. This is the table 1 show the information that collected from the questionnaire, and which is sex, breed, weight, color, and visit first time they visit the clinic or not. The first result we analyze is about based on objective 1, which is to determine the differences of CSS in different time point, and we found that CSS taken after 1 minute cat were placed in the cage is significantly higher compared to the CSS taken 20 minutes after first observation in the cage, with their p-value is 0.002. We use Wilcoxon sign range to compare this CSS between 2 times, which is after 1 minute cat were placed in the cage and after 20 minutes after the first observation in the cage. This result support to a previous study that CSS increased when cats were introduced to a novel environment but declined over time from the journal written by Gooding, 2012. The reason there are the increasing cats with the age because the cat may slowly to adapt to the novel environment and become more relaxed. Next result is based on objective 2, which is to determine the effect of cat individual characteristic on display of stress. First, we see the result for cat stress score after 1 minute placed in the solitary cage, and we found out that each have the significantly different with the cat stress score, and other factors were not significantly different with cat stress score. This is the table show the relationship between age and CSS after 1 minute the cat placed in the cage. Younger cats show significantly higher stress score compared to older cats after 1 minute after they placed in the cage. The p-value is 0.017. Then the result for 20 minutes after first observation placed in the cage, all the factors not show there are no significantly different between cat stress score. Previous study proved that there is a relationship between decreasing stress score and increasing age. Younger cat is more energetic compared to older cat, which make them score higher in a certain aspect in CSS, such as activity level. However, one study has found that there is a relationship between sex and CSS that we will not found in our study, which is stress level of mature male cat is higher than mature female cats. Next, we continue to approach test. We found out each was significantly different with approach test, and other factors which will not have a significant difference. This table shows the relationship between age and approach test. Older cats show significantly higher compared to younger cat in the approach test, which is the p-value is 0.049. From the table, we can see for approach test, cat more than 12 months will have a more count of approach than cats in less than 12 months.
Akila, you have less than 1 minute left.
Other cats show more approaching reaction to observer compared to younger cats that mostly show no approaching and retreat reaction. And lastly, for play test, there are no significant difference for 5 factors. And we also try to search the playtest latency, but no factor that significantly difference with the playtest latency. So the discussion the abnormal result for play actively were recorded in this experiment as the cat only show reaction approaching the toy and not approach. The cat may not feel safe or fully adapted to the environment to play actively with the toys. In conclusion, younger cats show more stress behavior during veterinary visit compared to older cats. Other factors affecting stress such as weight and sex may show the interaction between the stress behavior if more data were collected. Recommendation, we suggest to collect more data to get more significant result and choose the balanced number of male cats and female cats to obtain the strong interaction between sex and stress behavior. And lastly, we suggested to investigate if the nature status can give effect to the stress behavior. Is the list of reference that I used in the study. Thank you. That's all from me.
Thank you, Nur Akila. I have one question for you. Based on your result, you've mentioned that younger cats are showing a higher stress score, right? Do you have any idea why would that be? Why is that so?
It's because younger cat they is more energetic compared to other cat, which make them score higher in certain aspect in CSS, such as activity level, which is sleeping. Usually, older cat is sleeping or resting, but younger cat is more energetic, so CSS behavior will score higher in younger cats.
Yes, but how do you relate the younger cat being energetic with them being higher on stress score? How do
I
you relate
think
between the two? Like in a simpler words, why do you think the younger cat is much more is feeling the stresses more than the older cats? Kalau dia energetic, so what? Kenapa dia lebih stress kalau dia energetic macam tu?
because we do this study in the solitary cage, so the younger cat do not have space to run or escape from the observer or another cat in the veterinary care. So
Yeah.
they feel stressed more when they in the solitary cage.
Yes, I think that is the correct answer for that. Okay. Any more question from our panel?
One question from me. Okay. How can you ensure that the basal stress level for each of the cats tested in your studies are about similar, so that you're not taking biased data or something like that? Meaning you have accidentally introduced stress to the cat prior to the experiment. So how can you ensure that the stress level for each of the cat tested in your experiment are at the same level? Boleh faham tak that question?
Which mean if
Maksudnya before you start the experiment, okay.
Yes.
So the stress level of each of the cat that you tested to, how can you determine that they are similar at the same level? How can you score it? How can you determine? And during the experiment, the stress that happened to the cat is not it's not a carry forward from the previous state.
I'm not sure about that, but I only refer to this cat stress score body from their behavior, from body posture, stomach, leg, tail, head, eyes. From the after 1 minute, I place them in the cage.
I see. Okay, got it. Okay, thank you.
Okay, thank you, Dr. Kar, and thank you, Akila. Okay, you may stop presenting. Now we will move to our next presenter. We have Radin Muhammad Ajwad bin Muhammad Anas.
Alright. Can you see me, Doctor?
Yes, I can see you clearly.
Alright. Okay.
Smart hari ni nampak pakai tie semua. Dapat extra markah tu for the effort. Alright, you may share your slides with us.
Alright. Wait a minute. This. Alright. Can you see the slide?
It's coming. Okay, we can see your slide. Your time starts now.
Right. Assalamualaikum warahmatullahi taala wabarakatuh, and good afternoon to all the panelists and to my fellow course mate. My name is Radin Muhammad Ajwad bin Muhammad Anas, and I'll be presenting my thesis or research on domain analysis of uncharacterized proteins in Corynebacterium pseudotuberculosis, and I am supervised by Dr. Muhammad Fakrul Zaman Raja Yahya. Right. To introduce you to my research paper, Bioinformatics studies is a field of studies that falls under the studies of biology and computer science that incorporate the acquisition, storage, analysis, and transmission of biological data, especially in DNA, amino acid sequence. By computerization, bioinformatics develops software such as Basic Local Alignment Search Tool, which is BLAST in short, and Conserved Domain Database, which is CDD, to analyze the large data rapidly. This practice can be useful in retrieving the solution or in developing new therapy or way to treat diseases. In this research, however, the bacteria that will be used is named Corynebacterium pseudotuberculosis, which is an etiological agent for caseous lymphadenitis, which is a chronic disease in both superficial and visceral forms, which is characterized by swollen lymph node. The surface form occurs as peripheral lymph node enlargement and abscesses and typically affects goats and sheep. Right. The problem statement for my research is the cases lymphadenitis, which is CLA in short, disease caused by Corynebacterium pseudotuberculosis is severely affecting the goat and sheep industries worldwide. While many works highlight the CLA disease epidemiology, microbial resistance, and pathophysiology, there are still many Corynebacterium pseudotuberculosis proteins remain not well characterized. Now, what is the significance of my studies? This research could be beneficial for pharmaceutical scientists to develop new drug therapies or medicines, thus provide the potential solution against the Corynebacterium pseudotuberculosis threats. And the objective of the research is, of course, to analyze the functional domain of uncharacterized proteins in Corynebacterium pseudotuberculosis by using protein blast, which is BLASTP, and CDD. All right, how do I obtain the data and the result? There, there, that is why methodology comes, which is we done it first and foremost. 150 uncharacterized proteins of Corynebacterium pseudotuberculosis sequences were downloaded in FASTA format from the UniProt KB database. The uncharacterized protein sequence were later then used as inquiries in BLASTP for identification of homologous proteins. The parameters of search against the non-redundant database will be set to an e-value of less than of 1 exponential of -6, and the sequence identity acceptable will need to be higher than 30%. The protein sequences will then be analyzed using conserved domain database, which is CDD, for any associated domain, and then will later be analyzed again using Cello2Go server for additional information such as gene ontology and for their taxonomy. And all the findings will be recorded and visualized using pie charts and table figure, which will be presented right now. Right. Based on the research and the studies I have done, this is the pie chart that classify the protein sequence that I have analyzed based on their biological pathways. As you can see, there are a few and various biological pathways, and there are some that is specific, some that is general, and the major biological pathways, which is carbohydrate metabolism, which makes up of 10.7% of the sequence, is also the same as you can see from the pie chart, which is unknown, which is 10.7%, which agrees with previous research, which said that several genes found in this pathogen's genome have yet to be identified, and unknown is due to the random selection of the protein sequence data in the KB server. Right. Second most major biological pathways is the biosynthetic process, where the synthesize anything such as cell wall or even energy of some sort, which makes up of 8%. And the third most major biological pathways is involved in catabolic process, which makes up of 6.7% of the biological pathways. All right. On to the next pie chart, which is done by classifying the proteins by their molecular functions. Right. As you can see, there are various molecular functions, and just like the previous previous pie chart, they are unknown also, but this time it's less more percentage, and the major percentage of the molecular functions is dominated by DNA binding, which makes up of 21.3%, which makes sense because Corynebacterium pseudotuberculosis is a viral and an organism that needs replication and translation all the time and transcription. So DNA binding is absolutely the major molecular function of the protein sequences. Right. The second molecular functions of the protein sequences were dominated by kinase activity, which makes up of 11.3%, and the third most abundant molecular function is transmembrane transport activity, which makes up of 8.0% due to the molecule go in and out of the microorganism. Right, onto the third pie chart, which is done by classifying the proteins by their subcellular localization. Right, the protein will localize somewhere in the microorganism, and this is found that they have chosen at least one of the four localized place that have been found here, which is membrane, cytoplasmic, extracellular, and cell wall. As you can see here, the major localization is in the membrane, which makes up of 48.7%, which in numbers is 73 protein sequence which inhabit the membrane localization. And cytoplasmic is the second most abundant localized place, which makes up of 33.3%, which are 50 sequence in total, and extracellular only has 17.3%, which is 26 protein sequence, and cell wall is only one protein sequence, which makes up of 0.7%.
You have less than 1 more minute.
All right, on to the next and last pie chart, which is this is done by classifying the protein based on their domain that has been analyzed, and due to the various domain, I have not classified the domain to what kind of domain it is, but instead I classify it I classify it to is it does it have domain, or does it have domain that is known, and does it not? So 112 of the protein domain was found, which makes up of 75.3% of the protein sequence, and 38 protein was not found their domain, which makes up of 24.7%. Right. As additional infos, I also run it through TMHMM, which is the transmembrane server, and was the data was tabulated like so, and this is 10 out of the various data that I have. All right. As in a conclusion, prediction of transmembrane helices of Corynebacterium pseudotuberculosis is achieved by using Uniprot KB, BLASTP, CDD, TMHMM, and Cello2Go websites. The major biological pathways predicted are carbohydrate metabolism, biosynthetic process, and catabolic process. Major molecular functions were found to be DNA binding, kinase activity, and transmembrane transporter activity. The subcellular localization of the uncharacterized protein is mostly in the membrane, and among the 150 protein sequences, only 112 of the sequences has known domain, while 85 of the sequences contain transmembrane domain. It's all for me. Thank you. Yeah, sorry. As a recommendation, this species need more research as well as in-lab experiments to further specify the functions and pathways, because the limitation of this research is to do the various sequences which is yet to be reviewed and annotated, as it is still a new topic to be reviewed, and due to the limitation of ODL, no lab experiment can be done. All right, thank you. That's all from me.
Okay, thank you. Do we have any question from our panel? We have Dr. Diana and Dr. Karazwa.
Assalamualaikum, Radin. Can I ask you some question? Okay,
Waalaikumsalam, yes,
Radin.
betul.
How you calculate the sample size? I mean, how do you get 150 protein to be analyzed?
why do I use 150, the
Yes.
specific
How do you
numbers?
calculate the sample size?
Ah,
How do you get that? Why 150?
why 150? To be honest, I actually don't know why 150 because I just follow my supervisor advice to use 150 protein sequences as it is a lot already, and the total sequences that I can choose is actually around 7,326.
Yes, that's the reason you have to know how to calculate the sample size, because by having a main aim, then you know why and how 150 come. All right. Show me the result, please, Radin.
All right.
Show me the result, your result, the first slide of your result and discussion.
Right.
Okay, now talking about the protein. Okay, you already analyzed the protein and so on, based on the gene, okay, that you have. Okay, can you explain to me, since all the result and discussion, your result and discussion part didn't put any discussion, just a result. So can you tell me why most of the protein involved in carbohydrate metabolism pathway? Isn't it? Now you say, you tell us, you show to us that 10.7% of the gene is for the carbohydrate metabolism pathway, isn't it?
Yeah.
Okay, and then why? Why most of them are contribute or involved in the carbohydrate metabolism pathway, not the others pathway?
This is because carbohydrate metabolism refers to all the biochemical process that occurs in living things, organism to produce, break down, and into convert carbohydrates, which is done by microorganism to live, and that
Is
why.
it only the source? Is it
Yeah.
carbohydrate? Is it carbohydrate is only metabolite that you have in this organism?
No.
No. And then why carbohydrate metabolism become the most important thing here? Sebab you tak put any discussion here. That's a big
Yeah.
question there.
Actually.
Kenapa agak-agak carbohydrate metabolism yang penting? Sebab paling banyak tu.
Because it needs energy.
Okay, you are also talking about pathway, but here you only talk about the carbohydrate. The big question is, how about the protein? How about the lipid metabolism? It's almost absent here. What can I say? Based on your pie chart, is almost absent. So that's the reason I ask you why you choose for 150. Maybe mungkin itu yang your supervisor aim for. Itu yang you kena discuss with your supervisor. That's the reason. Then the answer maybe help you to answer my question. Why most of the gene involved in the carbohydrate metabolism? Okay.
All right.
It's okay. You just discuss with your supervisor. Okay.
I understand. Thank you, Doctor.
Okay, welcome.
Alright, thank you Radin Ajwat. Thank you Dr. Nurdiana. Alright, without further ado, okay, sorry ya saya terpaksa laju because we have to keep our schedule on time.
Alright.
Our next presenter is Nur Ilya Najiha binti Nasir. Ilya, dah ready?
Yes, Doctor.
Alright.
Boleh nampak slide saya?
Yes, we can see your slide. Okay, you may begin and your time starts now.
Okay, Assalamualaikum and good afternoon. Good afternoon to all the panelists and fellow friends. So my name is Nur Ilya Najiha binti Nasir. I have been supervised by Dr. Azani Saleh, and today I will present my article review titled In Vitro Regeneration of Oil Palm. So this will be my presentation outline. As introduction, Elaeis guineensis, or known as African oil palm, originated from West Africa. Oil palm known as a high demand source for food production and biofuel due to the potential of the plant in producing 7 times more productive oil compared to the best yields of other oil crops. Oil palm also can produce 2 main products, which are kernel oil and palm oil. So economic values, oil palm production occupied 4th major contributor to the Malaysian economy. Besides, included among the major contributors to Malaysia's GDP, earnings in foreign exchange, and the existence of employment opportunities. So this is my 5 research questions. First, what are the aims of this review article? Second, is plant growth regulators or PGRs give positive effects on plant tissue culture? Third, is the concentration of PGRs directly correlated with the growth of plantlets? Fourth, is the addition of PGRs will determine the success of acclimatization? And fifth, is PGRs will give the same effect to the different type of explants. My research hypothesis, the explant cultured on culture media supplemented with suitable plant growth regulators will promote more shoot and root formation compared to the plant cultured on unsuitable PGRs. So problem statements on why oil palm need to be cultured by in vitro regeneration. This is due to the low rate of seed germination if we use traditional method. So the objective of this review article is to identify the effects of plant growth regulators on in vitro regeneration of oil palm. A bit on in vitro regeneration of oil palm. In vitro regeneration defined as a technique where explants forming organs and tissues after undergo cell division and differentiation. In vitro regeneration can be performed in 2 ways, which are somatic embryogenesis and organogenesis. And through in vitro regeneration, this can provide advantages compared to traditional seed propagation method. There are several factors affecting success of in vitro regeneration, and in my article review, I will focus more on the effect of plant growth regulators. So plant growth regulators defined as the natural or synthetic biological substances that affecting plant growth and development. There are several classes of PGRs, and the commonly used are cytokinins and auxins. So first, root induction. In the root induction process, a new root will be grown with the aid of auxin growth regulators because auxin will influence the process of cell division as well as root formation. And in general, the rooting phase occurred right after the first 30 days of culture, and up to 3 months, the best rooting will be obtained. So findings from my article review is first, Currently et al. 2015 stated that the oil palm shoots used were produced by somatic embryogenesis. Thus, only the number of fruit of the explant treated by NAA increased from the 4th week up to the 7th week with an average number of shoot 3.26, while IBA and IAA showed no significant difference from the control which no auxin added. And second, by Nizam and Tikato 2009, studies showed that media supplemented with 6 mg per liter NAA and 9 mg per liter PBZ produced the highest rooting percentage of fibrous root system. For multiple shoot formation, research made by Nizam and Tikato 2009, 3 types of culture media were used, which are half MS, MS, and WPM. This media then supplemented with 2 types of PGRs, which are NAA and PBZ, with different kind of concentration. So as a result, compared to half MS and MS, WPM medium, which woody plant medium, supplemented with 6 mg per liter NAA and 9 mg per liter PBZ indicate the most suitable combination, which produces the maximum number of shoots, 2 shoots per explant, with length of 11.4 cm. For acclimatization, the condition where in vitro plants are then transferred and adapted to the new environmental condition. So previous study by Currently et al. 2015, through mycorrhizal applications, the attachment of mycorrhizal spores and the hyphae form symbiosis with the new roots of oil palm. Therefore, 4 different doses treatment of mycorrhizae towards oil palm shoot treated during in vitro has been tested, and it has been found that 4 grams of mycorrhizae per polybag showed the highest hyphal infection percentage compared to other doses. So this resulting in different growth pattern that can be observed between mycorrhizal treated seeds and those without mycorrhizal treatment. And last, somatic embryogenesis. Somatic embryogenesis is developmental process of plant somatic cell that differentiate into totipotent embryonic cell that has ability to give rise to an embryo. So a study conducted by Yusnita et al. 2011 using young leaf segment as explant, induction of embryogenesis was conducted on MS medium supplemented with 450 micromolar of 2,4-D, 18 milligram per liter of adenine, 4.4 micromolar of benzyl adenine together with activated charcoal. So this resulting in 10.8% of clump callus became embryogenic after 12 to 18 weeks. And according to the researchers, in order to achieve the development and maturation of somatic embryos, the embryogenic callus then transferred into EDM1 and EDM2. So as a result, they stated that EDM1, which contained MS medium supplemented with 30 gram per liter sucrose and 2 gram per liter activated charcoal, was better than EDM2 for somatic embryos development. So in conclusion, NAA was more effective in inducing roots formation for in vitro regeneration of oil palm. The formation of roots then can be enhanced with the presence of PBZ. And the combination of NAA and PBZ was also effective in inducing shoot formation of oil palm. These are my 3 recommendations toward further studies. First, although the effects of plant growth regulators have been widely studied and identified, other types of PGR such as dicamba, TDZ, and etc. could be tested. Second, more research on acclimatization of oil palm need to be conducted, such as the effects when transferred into coconut fiber, sand, hydroponic system, and soil. And third, further research on other supplements such as coconut water on the enhancement of in vitro regeneration of oil palm could be conducted. This is my list of references. And that's all from me. Thank you.
Thank you. Thank you. Thank you, Nur Ilya. All right, you are the first student who managed to finish your presentation on time, bang on time. All right, thank you very much. I would like to open the floor for a question and answer session. Do we have any question from the panel?
Ilya, Assalamualaikum.
Waalaikumsalam.
Okay, just would like to ask you a simple question. Okay, can you show me your aims of your review? Okay. All right. Now, based on your objective or your aims, does you compare with the traditional approach? Does you compare your review, okay, with the traditional approach?
Okay, before I choose this plan, I have identified that using traditional method, several months are needed in order to grow a single plant.
Okay, have you mentioned about the disadvantage and advantages in your review? Sebab kat sini I tak nampak.
Dekat
Means
slide
in your
tak ada,
written.
tapi in written ada.
Yes, in the slide tak ada. In detail ada.
Yeah.
Then you have to mention about this, okay? Because to achieve your aims, okay, you need clearly to identify what is the advantages and also disadvantages comparing between these two approach. Because by having that point, you can come into your conclusion that this technique is good compared to what thing. So bila you cakap bagus je, banding apa? How good that your suggestion pun you have to justify. That's the reason in the slide you should tell us advantages and disadvantages. You make a critical comparison between these two approaches. Okay, so in your return, I hope, since I am the second examiner, I hope I can have and I expect to read the advantages and disadvantages from both techniques. Okay, Ilya?
Okay, Doctor.
Thank you, Dr. Diana. Dr. Kar, do you have any question for Nur Ilya?
Okay, thank you, Dr. Rosnida. Okay, Elia, okay, you did mention that you use coconut water, right? As
Yes.
part of the medium that you use. Okay, so my question is, how can you ensure that the different batch of coconut water that you use are similar in terms of nutrient content in it?
Sorry, doctor, because I stated the coconut water in my recommendation. Is that what you mean?
Oh, recommendation, I see. It's not,
Yes.
are you using it? Do you use it?
No.
No. But just for general question for you. So, how do you think that we can ensure that the nutrient content, okay, in the coconut water in different batch of coconut are similar?
Because
Otherwise, the medium that you produce will be slightly different in terms of nutrient, right? In terms of the chemicals that are in the medium.
based on my readings that and guide from my supervisor, she stated that coconut water can act similarly as plant growth regulators because coconut water itself rich in lysine, cysteine and other amino acids, and also rich in carnitine type of cytokinin which can promote cell division and growth.
So you're talking about hormones there, right? But
Yeah,
how can you
hormones.
ensure that the level of hormones or the concentration of hormones are similar in that particular different batch of coconut water?
Sorry, I don't think I have the answer for your questions, but I can read several journal.
Yeah, please. Thank you. If you can include it in your write-up.
Okay, thank you, Doctor.
Cool.
Okay, thank you, Dr. Karasfa, and thank you, Nur Ilya. Okay, let's move on to our next presenter. We have Nur Shafiza binti Rusidul Bukhari.
Thank you, Dr. Asmida.
You're welcome. Nur Shafiza?
Yes, I'm in here.
If you can start sharing your slides.
Will like sekejap eh.
Do you have problem with your slides?
Yes, tapi sekejap eh. Can you see my slide?
Not yet coming.
See?
Yes, we can see your slide now. Okay, you have 8 minutes. Okay, your time starts now.
Assalamualaikum and good afternoon. I bid to all panel examiners and to my FYP colleague. My name is Nur Shafiza binti Rusidul Bukhari and I will present my thesis with the title of Entomofaunal Diversity of Class Insecta at Selected Forest in Pulau Tuba. Entomology is the study of insects that involve their morphology, behavior, evolution, ecology, biodiversity, and as well as their role in the ecosystem. It is estimated that 1 million species have been described, and according to Campbell et al., insects are classified into 30 orders with 8 dominant orders. This study was conducted at Pulau Tuba Forest near Gua Kelawar and Bukit Putih. The island was located 5 kilometers southwest of Kuah Jetty, and it takes for 50 minutes to 20 minutes to Pulau Tuba. This island is called Pulau Tuba because of a native plant species named Tuba plant that can be found abundantly in this island. The objective of this study was to determine the diversity and abundance of class Insecta at selected sampling location in Pulau Tuba, and to differentiate the diversity and abundance of class Insecta at 3 different locations ranging from forest fringe to the inner forest. In this slide, there was 4 methods that were used. First, the malastrap was set up every 100 meters for 3 units from 3 different locations of forest, which were forest fringe, middle forest, and inner forest. Second, the sampling collection was conducted by Dr. Siti Khairiyah Muhammad Hatta from 17 September to 21st September 2020 in Pulau Tuba. Third, the order identification was conducted by us at the UiTM laboratory, which we sorted the insect according back to the morphology and physical appearance, and then the sample of insect, we pin the insect using the entomological pins and was dried up at the temperature of 38 to 40 degrees Celsius. Last but not least, the data was analyzed by using SPSS and FAST software to measure the Kruskal-Wallis hash test and to measure the indices of Shannon-Wheeler diversity index, evenness index, and Magalhaes richness index. For the overall result, the total individuals collected was 5,883 individuals, with 106 morphospecies identified and 7 orders were recorded. The 7 orders recorded was Datudia, Coleoptera, Diptera, Hemiptera, Hymenoptera, Isoptera, and Lepidoptera. Figure 1 show that the highest number of order is order Diptera with 93%, followed by Hymenoptera with 3% and Hemiptera with 2%. The least abundance of insect or order in the Pulau Tuba is Latodia and Isoptera with 0% respectively. Based on table 1, abundance and diversity of insect, each tract represents different location of forest, which were tract 1 represent the forest fringe, tract 2 middle forest, tract 3 represent the inner forest. The distribution of orders between tracts show that tract 1 has the highest number of total individuals with 2,817 individuals with 78 morphospecies, followed by tract 2 with 2,592 total individuals with 18 morphospecies, and lastly tract 3 with 474 individuals with only 10 morphospecies. The most abundant order collected was order Diptera with 5,450 individuals across all tracts, and the most diverse order was Hymenoptera with 32 morphospecies recorded. The least abundance and species richness was order Datudia with only 6 individuals and 1 morphospecies only. Each tract has only 6 orders out of 7 orders, which means that the diversity of insect at Pulau Tuba is low, but the abundance of insect in each tract has a higher number. This is due to rainy season and short period of sampling collection of insects. Based on table 2, tract 3 has the highest value of Shannon-Wiener diversity index with 0.53, evenness index 0.28, and the Margalef richness index with 0.81 value compared to track 1 and track 2. For the overall, track 3 was the most diverse, least dominance of species, and has the highest species richness compared to track 1 and track 2, even though the number of individuals at track 3 was low. According to Muhammad Hatta et al., this is due to the location of track 3, which in the inner forest that was away from human activity areas and have high humidity and low temperature. For the conclusion, the null hypothesis was rejected due to the p-value less than 0.05 and there was a significant difference between order across categories the tracks. The significant difference comes from two pairs of tracks, which were track 1, track 2, and track 2, track 3. And there was no significant difference between order distribution for track 1 and track 3. For my recommendation, further studies are needed to be conducted at Pulau Tuba to get more data about diversity and abundance of insects. Besides that, a longer period of sampling collection also needed because to obtain more accurate data. And lastly, we required to identify the insect using the insect collection from Center of Insect Systematic, but due to the pandemic and the center was closed, that is difficult for us to identify the insect as a reference. That's all for me. Thank you.
Thank you, Nur Shafiza. Okay, sama juga, right on time, good. I have a few questions for you. Okay, first of all, can you tell us what does it mean by morphospecies?
Morphospecies, we differentiate their physical appearance. Means that the difference of wings, difference of their legs with the other species. That is the morphospecies.
Thank you, Nur Shafiza. My second question is, okay, before I go to my second question, is it possible for you, instead of putting in the word morphospecies, why it is not possible to put it as species? Species sahaja.
Because some species have the different physical appearance. So to sort the insect, we need to sort them using the morphospecies, such as their different wings, their body color, and so on.
All right, that will bring me to my second question. You've mentioned just now that Diptera is the highest order when it comes to the number of individuals, right? You are referring to number of individuals?
Yes.
Okay, so in a conclusion, what is the significance of having Diptera as the highest individual?
The significance.
Yes. Like you have done all of this study, okay, and then okay, you do study, and then you should have a bigger picture of what this study is all about. Based on all of the data that you have managed to collect, macam okay, you've mentioned that the highest order would be Diptera. So what can you say about the Pulau Tuba when it comes to diphtheria? Apa kesan dia bila diphtheria is the highest number of individual, maksudnya paling banyak diphtheria within that area. So what can you say about the area?
That means that at the Pulau Tuba, the species that was abundant is Diphthera compared to other order. And Diphthera is mostly in other papers that I read was the mostly abundant in the mostly abundant order or in the other area.
I don't think you have. I think what you are doing is your supervisor collect the data for you, betul kan? Dr. Kai yang pergi field kan? Okay, she collect the data for you, and then you're the one who analyze the results. Okay, based on the percentage, based on the indices tadi kan? Tapi you have to have you should know the reason why your supervisor asked you to do all of that. Kita bukan simply macam collect data and then kita cakap okay, species ni paling tinggi, species ni paling rendah, order ni paling tinggi, order paling rendah. You have to understand why you are doing that. Okay, faham? So nanti duduk dan fikir kenapa you buat kajian ni. That will answer my question, right?
Okay.
Alright. Okay, since we do not have enough time on our side, okay, we will move on to our next presenter. Thank you, Nur Shafiza. Our next presenter is Munira binti Muhammad Sazali.
Hello, can you hear my voice?
Yes, we can hear you, Munira. You can share your slide.
Can you see my slide?
Yes, we can see your slide. You will be given 8 minutes. Your time starts now.
Assalamualaikum warahmatullahi wabarakatuh to all lecturers and friends. My name is Munira binti Muhammad Sazali, and today I would like to present my final year project titled Morphological and Molecular Identification of Culturable Cyanobacteria from a Public School Water Feature. My presentation will cover all these topics. But first of all, I want to introduce to you guys what is cyanobacteria. Cyanobacteria are known as blue-green algae, which are gram-negative bacteria and develop oxygenic photosynthetic, where they use sunlight for growth, and photosynthetic pigments for energy. They usually appear and originated in freshwater and also terrestrial ecosystem. According to Faldo et al. 2014, cyanobacteria are categorized based on their morphology, cellular differentiation, biochemical, physiological, and genetic characteristics. The cysteine-S RDNA gene is found in all bacteria, where it is the most informative gene for studying ancestral relatedness. Cysteine-S ribosomal RNA gene encodes the RNA component of the bacterial ribosome 30S subunit. By far, the most commonly used tool for studying bacterial phylogeny and genus grouping is cysteine-S RDNA gene sequencing. Cyanobacteria can be classified based on their morphology and grouped into 5 orders. There are 2 orders in filamentous shape and 3 orders in cocoid shape. Morphology of cyanobacteria can be changed due to the environmental factors and the stage of development. Some cyanobacteria can show high morphological variability under different growth conditions, making identification of species based on morphology alone are unreliable. For example, in one of the papers produced by Han et al. 2017, the isolate cyanobacteria grew as a dark blue-green filaments in the medium culture, while on solid media, it grew in colonies. To overcome this problem in cyanobacteria, molecular techniques have been introduced to identify cyanobacteria, which is using cysteine-S RDNA gene sequencing. The objective of my research is to identify the cyanobacteria using cysteine-S RDNA gene sequencing and to compare between morphology of characteristic with the cysteine-S RDNA sequencing result. The finding of this study provided information on the identification of Cynobacteria using 16S rDNA. Furthermore, the misconception in identifying Cynobacteria urged this study to be conducted using molecular analysis, which is 16S rDNA gene sequencing to get an accurate result and more depth information on Cynobacteria in Malaysia. Next, for methodology. For microscopy identification, the morphology was observed through microscope. The individual cells were observed at 1,000 times magnification. Trichomes were observed and cell length were observed roughly based on the observation. Next, there are several steps in molecular identification. The first one is DNA extraction, where easy pure bacteria genomic DNA kit was used to obtain high-quality DNA sample from cyanobacteria isolates. Then, electrophoresis was done in 0.8% agarose gel for 30 minutes. Quantity of DNA was estimated spectrophotometrically by measuring absorbance at 280 nanometer, while the quality was determined by taking ratio of absorbance at 260 and 280 nanometer using a biophotometer appendage. In order to get the sequence of 16S rDNA from cyanobacteria isolates, the 16S rDNA gene was amplified using two primers, which is CR106F and CR781R. To check the presence of PCR product, gel electrophoresis was done in 1.5% of agarose gel. After purification using Kia Quick PCR purification kit DNA sequence data was obtained and analyzed by BLAST. This is the result for microscopy identification. From the figure, the cyanobacteria isolate meet the characteristic features under oscillatorialities order. This isolate was characterized by being filamentous and green in color. The strain shows that the length differed from each other. The cell also located in a group. Some of them are unable to form branches. Oscillatoria usually form a mucilaginous sheath. Next, the cells are multicellular and having a reproduction by binary fission. Their vegetative cells are in root shape with having diameters from 1.0 until 3.0 micrometer. All members of oscillatorialis do not form heterosis. Heterosis is a specialized nitrogen-fixing cell formed during nitrogen starvation by some filamentous cyanobacteria. This is the result for gel electrophoresis of PCR. The 16S rDNA gene was amplified using this PCR. From the observation, it is proved that DNA has been successfully amplified. DNA ladder used was 1 kb DNA marker, and the gene product observed was 600 base pair inside as seen in agarose gel electrophoresis. The markers band were readily visible from at length 6, the absence of DNA bands in the negative control showed that the reagents were not contaminated by DNA, while at length 7 there is one band shown where it is the good process for it is the good result for this process. Next, this is the result for BLAST analysis. Based on the BLAST study of 16S rDNA sequence homology, this is the top 5 hits closest species. The highest hit is species Cyanobacterium species PBJ2 and PBJ1 with the percentage identity of 99.00%, and the rest is Leptolimbia species and uncultured bacterium. Based on NCBI taxonomy database, Cynobacterium species PBJ2 was classified under the order of Oscillatorialis. Thus, it concluded that morphological characteristics and molecular characterization can depend on each other to get an accurate identification. In conclusion, the isolate sample have similar characteristics under the order of Oscillatorialis. Thus, identification of cyanobacteria through morphology and the result from BLAST is in line with 16S rDNA gene sequencing result. For the recommendation, the application of modern molecular analytical approach such as in the characterization of cyanobacteria. This is the list of references I use in this presentation. That's all from me. Thank you.
Thank you, Munira. can I have a look at your title once again? Okay, you are doing the molecular identification to match the morphological identification. Am I right?
Yes.
Okay, so based on that,
Yes, Doctor.
you have your samples, okay, a few species from Oscillatorialis, and then you name them,
Yes.
you identify them morphologically, and then you match with the molecular. So how sure
Yes.
can you show me the result once again? Yang ada percentage, okay, kejap nanti tengok dia. Tengok yang tadi kejap. Ini oscillatory, okay. Yang percentage tu.
Percentage plus analysis.
This one. Yes, blast analysis. Okay, based on this one, okay, you've mentioned that you have PBJ2, PBJ1 dan semua tu lah kan? So all of this has not been identified morphologically, right? Yang ini,
Yes.
has the sample been identified morphologically?
I think yes. When others want to check whether their morphology same with the molecular, they can just use plus analysis.
So you are not doing this analysis yourself. You get the result from somewhere else. Is that what you meant?
No, I plus this on myself. I get this sequence from my supervisor, and then I plus
Okay.
this on myself.
Tapi that sequence tu, okay, is it coming from the samples on which you have been using it for morphological identification?
Yes.
Kejap, I'm a bit confused here. Tengok lagi result yang selepas tu.
This one.
Lagi?
This is the jet electrophoresis.
Okay. Okay. Based on your results, you have not mentioned, like you are focusing on the morphological identification on which, okay, your title is on morphological and molecular molecular identification. However, you failed to show us, okay, how does it differ or or where the similarities are. For example, when you have the samples, okay, you should be identifying it morphologically macam you cakap lah. You look at the structure, okay, either it is the shape lah, rod in shape ke, spirals ke, and then you look at all the reproductive structure semua tu kan, how macam dia fragmentation ke, dia ada branching, tak ada branching, and then you name them based on that, okay, and then when it comes to morphological, when it comes to molecular identification, we put it in blast, and then memang terus keluar nama yang tu lah, yang ulang balik yang percentage tu, yang blast analysis tu. Memang terus keluar. Okay, let's say you have that sample on which you already name it Cynobacterium sp tu. Masa you buat yang morphological identification, memang nama yang you bagi Cynobacterium sp.
No, actually the morphological meet the characteristic Oscillatorialis, and this Cynobacterium species PBJ2 is under Oscillatorialis order also.
Oh, so in that case, you are not able to identify lah sample tu species apa kan? Morphologically. Betul tak?
Yes.
Okay, now I understand. Because usually, okay, because Oscillatorialis is quite common, okay, for freshwater algae. Ataupun in this case, you buat sebagai you regard it as bacteria lah, cyanobacteria. Tapi sebab saya bidang phycology, so kami panggil we call it Cynophyta. Okay, so instead of cyanobacteria, kita panggil Cynophyta. So usually, okay, morphologically kita dah tahu dia species apa. Okay, kita dah tahu genus apalah, in this case genus apa. And then sometimes, because if it is very common, kita terus boleh bagi sampai ke nama spesies without proceeding further with the morphological analysis. Tapi because you have the resources, boleh je buat dua-dua to confirm. Okay, kita namakan dulu gunakan morphological characteristic, and then to confirm the species yang kita dah bagi nama tadi tu, okay, we can use molecular analysis, okay, to confirm nama species tu betul tak? Macam tu, right? Faham? Panjang saya tanya ni. Do we have any question from other panels, Dr. Diana dan Dr. Khai?
Just to add a simple suggestion, buka balik dekat morphological diagram tu, picture. Okay. Since you observe this sample under microscope, the first thing, sebab saya dah nampak tadi ada dua student yang buat the same mistake, you need to add ruler. Okay, you need to mention about the scale. What is the magnification? This is talking about the microscopic identification. So please add the ruler that describe the magnification of the observation, okay, in your sample.
I'm sorry because due to limitation entering lab, my supervisor
Kita boleh masukkan dalam, okay, Munira, we have free access dekat internet je, you Google saja, we can add manually. Okay, I tahu you tak boleh masuk lab, that's the reason. Okay, so you just add, you dah tahu kan that what is the magnification used, okay, and then you add the magnification on the tool, okay, and then you can get the ruler on your image.
Okay.
This is how to present your microscopic image. Okay, and then in your thesis, in your report, please mention clearly about the magnification. And how you get 1.0 to 3.0 mm, how you calculate this thing, the diameter?
This is just an assume because I just took from the oscillatorialis, the information from oscillatorialis order.
Sebab I nampak kat sini satu, dia tak ada dia punya standard error semua tak ada. And then if you calculate, you may use image J. This is just my suggestion. You may discuss with your supervisor. Image J, this is also the tool, free tool. Okay, you can use that thing. Okay, you insert your image and then you can calculate the size of the sample. Okay, the region that you are targeting for. Alright, that's all from
me. Thank you, Dr. Diana. Tapi Munira, you tak ukur sendiri kan?
Tak.
So this diameter that you have and all of the description that you've mentioned here bukan datang daripada your study kan? You daripada literature kan?
Yes.
So this is not supposed to be in result and discussion. Sebab benda yang kita masukkan result and discussion adalah data yang kita analisis atau data yang kita collect. Macam if you are looking at if you would like to look at the diameter of the oscillatorialis, okay, you kena ada the samples of species under oscillatorialis and then you measure them. And then baru you boleh buat, okay, it ranges from berapa millimeter to berapa millimeter. Let's say you have 5 samples, okay, of different species ataupun probably different genus ke kan. Then you measure them, baru you boleh buat, okay, ranges from berapa to berapa. But because you have not measured them, you tak boleh lah mention kat sini. Okay, this is supposed to be in the introduction or literature review macam tu. Okay. One more thing is because you are looking at oscillatorialis and we have different species under oscillatorialis and your image is very blurry. Is this your very own image ke? You Google ke?
No,
punya kan?
I got it from my supervisor.
Okay, so this is real samples lah. Alright, yeah. The thing is it is blurry. Kita tak nampak apa pun. And when you label it as oscillatorialis, you're supposed to be seeing, okay, different structures here on which representing different species under oscillatorialis. Ada banyak species under oscillatorialis as you mentioned just now. Your sample sendiri pun ada 5 yang you bagi tadi kan, and apart from that we have so many other common oscillatorials in freshwater. So mungkin you boleh Google gambar tu, just name them so that examiner will have an idea what types of species represented by oscillatorialis ni. So you keep mentioning we keep mentioning Oscillatorales tapi kita tak tahu apa spesies apa dalam tu. So at least give a few yang very common lah. Okay, just to give an idea to the examiner, dia nampak oh ni macam ni nostoc, oh macam ni apa ni limbia, macam ni apa-apalah spesies yang ada Oscillatorales, especially the Oscillatoria itself. Okay, sebab ada satu spesies ada nama Oscillatoria tu, dia memang ada Oscillatoriales. That is the common ones for Oscillatoriales. Okay, faham?
Okay, thank you doktor.
Alright, you're welcome. Dr. Kah ada apa-apa nak tambah ke?
Dah tak sampai. It's okay.
Okay, that's fine. Alright.
Okay, cool.
Okay, we will move on to, thank you Munira. We will move on to our next presenter. Kita dah 40 dah sekarang. Okay, we have a former presenter, Nurul Ain Farhana. Nurul Ain ada? Okay, Nurul Ain, you may share your slides now. Ada problem ke Ain? Okay, it's coming.
Okay.
Okay, we can see your slide. You may begin now.
Okay. Assalamualaikum and good evening. My name is Nurul Ain Farhana binti Saleh, and today I will present my final year report project. And the title is a 3 years retrospective study on the Clinico-Laboratory data for human papillomavirus infection from private healthcare KPG hospitals. So HPV stands for the human papillomavirus that sexually transmitted infection associated to the cervical cancer. So today, based on the record of the KPG hospitals in Malaysia, there are 19 high-risk genotypes associated with the high-risk infection, and there are 9 genotypes associated to the low-risk infection. So among these high-risk genotypes type 16 and 18 has predominantly associated with the cause of the cervical cancer, and low-risk HPV 6 and 11 predominantly associated with the cause of the anogenital warts. So the study has been carried out by 3 main objectives. First, to analyze the demographic characteristic of the patient positive for HPV high risk and low risk by the multiple real-time PCR in KPG hospital. Second, to determine the diversity of the HPV genotypes according to the low risk and high risk types of the infection and the cytology findings reported in KPG hospitals. And lastly, to determine the temporal distribution of the HPV infection by month from 2015 until 2017. So HPV is the small virus without an envelope and has been double-stranded viral genome. And today, here is the high risk and the low risk genotype that, as I mentioned before. So the common symptom, which is cell lesion, as shown here, it could be a wart like a growth in vagina or bumps that shape like cauliflower at the genital area. So in terms of the prevalence, worldwide, according to the National Center of the Biotechnology Information 2019, HPV 16 and also HPV 18 prevalence genotypes of high risk infection, whereas for the low risk is HPV 6 and also HPV 11. In Malaysia, a study that being carried out by the Hamzi in 2014, HPV 16 and 18 are the prevalence and is dominated by the Chinese races, followed by Malays and then the Indians. Whereas for the low risk, HPV genotype is 6 and also 11. So, since this study has been carried out on 7 years ago, yes? Yes? Yeah, doctor.
And okay, proceed with your presentation. I think something wrong with the connection. Okay. Continue with your presentation.
Okay. Okay. So as I mentioned before, since this study has been carried on 7 years ago, so using data from the 2015 until 2017 as part of the public health surveillance, we need to update our knowledge, information about the current circulating HPV genotypes in our country. So therefore, one of the main source of the academic information, the data we can get from hospital, such as private hospital, KPJ. So KPJ is one of the biggest private healthcare and its community of Malaysia with 25 hospital branches throughout the Malaysia. So to achieve the three objectives mentioned before, retrospective study has been carried on, and this study by using the PCR data that has been conducted by the KPJ hospitals from 2015 until 2017. So this PCR actually targeted the 19 high-risk HPV and the 9 low-risk HPV. So I analyzed the secondary data for the demographic characteristic and all the specimen originating from all 25 hospital branches categorized into 5 different regions. So the data I analyzed by using the SPSS for demographic profiling, which is HPV distribution, HPV positively frequency, and also the cytology result. So the result answer the first objective, which shown that the sample origin was subjected to the high risk and low risk HPV testing in KPJ, and all of them were women that being tested whether have positive HPV or not. So this table show that the most suspected region that have HPV cases at the southern and also the west region. So this slide shows that the number of patients that have positive HPV result from 2015 until 2017, which has 570 cases, and most of them caused by detected at the southern and also the central region of the Malaysia. So based on the second objective mentioned before, the result shows that the most prevalent high-risk HPV genotype in Malaysia based on KPJ record for the 3 years, which HPV 52 with 41 cases, and the low-risk HPV is HPV type 6 with 25 cases. So based on the data given, there are 3 result finding at the HPV 52, which is first is ascus. Ascus is an abnormal cell in the tissue that line with the outer part of the cervix, and is abnormal cell that we get from yeast infection. So ASCH, which can progress to the cervical cancer if left untreated. So LSIL is a frequent abnormal result in pap test that classified under the mild dysplasia and caused by the HPV infection that generally can resolve on its own. So for the type 6, the specimen from the female patient may also known as the reactive cellular alteration, which the way cervical cell appear when there is infection or the inflammation present. So when patient have the fungal infection, which consists of the Candida species, vaginitis might occur and can lead to the pain, itching or discomfort when during the sexual activity. So as I mentioned before, the most common HPV in Malaysia was 16 and 18. But this study shows that the current result, which the most prevalent high-risk HPV is 52. So KPJ, as the biggest private healthcare, and we get the secondary lab data from KPJ, can represent all the circulating genotypes in Malaysia. So in addition, according to the NIH 2021, China also one of the country that reported HPV 52 was the most prevalent HPV type in their country. So all the nationwide been talking about HPV 16 and also 18, but actually HPV 52 is the emerging HPV genotypes in nationwide. So next is number of patients diagnosed with a dual multiple infection of HPV genotype. So dual and multiple infection were found in 91 out of 570 cases, which dual infection more prevalent than multiple infection. So in 2015, no cases can be detected related to the dual or multiple infection. But for 2016, dual infection can be detected, which is a combination of HPV 6 and 70, vaginal intraperitoneal neoplasia, are the precancer cells found in the vagina. So this is a dual infection, HPV 52 and 58, and these are multiple infection.
1 minute. You have less than 1 minute.
Okay. Multiple infection, HPV 16, 39, and 53. So this is the temporal distribution of HPV infection by month. And so in conclusion, 570 patient, which is women, have likely to have get the HPV 52 for high risk genotype and HPV 6 for low risk genotype compared to the worldwide that mostly have HPV 16 and 18 as the most prevalent high risk. So as now there's two most common vaccine, which are Cervarix and also Gardasil, to prevent infection with just two high risk HPV, which is 16 and 18. So I recommend that scientists need to develop new vaccine to prevent the spread of the other genotype of HPV. That's all for me. Thank you for listening.
Thank you, Nur Ain. Okay, do we have any question from Dr. Diana or Dr. Ka?
Nurul Ain. Okay,
Dr. Ka,
I would like to
tanya
know
tu.
the Asfa. Hello.
What? Yeah.
Can you hear me?
Yeah. I can hear
Okay,
you.
great. Okay. So this HPV, okay, is it a DNA virus or RNA virus? And what is the rate of mutation?
The HPV is a DNA virus, and the rate of mutation is quite high because HPV is number 4 common disease in Malaysia and nationwide.
Quite high in terms of percentage. What is the number of the prevalence of mutation?
It depends of the type of HPV. Actually, if we talk about the cervical cancer, it's more than 70%. HPV can cause the cervical cancer, so it's quite high. And HPV also can cause like anal cancer, penis cancer, something like that, with different type of the genotypes.
Okay, thank you.
Thank you,
Okay.
Dr. Ka. Dr. Diana?
Okay, Nurul Ain, I would like to ask you some question. Okay, you have emphasized that HPV 72 is the emerging type, isn't it?
52.
Yes, 52. Sorry,
Okay.
52 is the emerging type. So, based on your finding on 520 patients, isn't it? The data collected
570.
70. Okay, 570. Can you recommend? Okay, why? Can you elaborate or explain to us why this vaccine is not available until here? Since you are is mentioned about the emerging type as an emerging type.
Okay. Actually, HPV, as we know, HPV 52 is the high risk HPV genotype. So, in nationwide, most HPV genotypes, the prevalence is 18 and 16. So, they use the same vaccine as 18 and 16 to cover the other genotypes of high risk and low risk HPV.
But why this problem is not overcome yet?
Because the symptom of the HPV 52 is not very visible, and it in 1 or 2 years it can resolve by its own. So there's I have some research. They said maybe
What do you mean by
the
dissolve? What do you mean by dissolve?
yes
You say that
resolve
dissolve.
resolve
Okay, resolve.
resolve by
So
its own.
okay, resolve by their own. So do you need? Do you think that we need a specific vaccine for them?
Yes, I think we need a specific because if we cannot prevent it earlier, it can cause the cervical cancer also.
But you say that you say before that the symptom appear at the least at the late stage. So
Yeah.
how can we prevent that thing? How can you solve that thing?
Actually, we need to go to hospital and do the but uh, we need to go to hospital and do the what we call diagnose for the early stage of the HPV. As a woman,
Yes, okay. When we're talking about the early stage of the HPV screening, okay,
yeah.
at that stage we can't identify the HPV 52 yet, right? Am I right?
Yes, yes,
Yes.
yes.
So how can we, when you say that you need to have the vaccine for 52 specific vaccine, means specific vaccine. So how can you determine who are the person that you need to give this vaccine since this come for the late stage?
Oh, um, uh, can you repeat your question, doctor? I confused right now.
Okay, sekarang ni, okay. You kata benda tu datang lambat. Okay, I make
Yeah,
it
yeah.
simple. Okay, you kata the symptom appear at the late stage. Okay, isn't it?
Yes, yes,
Okay,
yes.
now you are talking about, you are suggesting that we need to develop a specific vaccine for HPV 52.
Yes.
So my question is, how do you recognize which person that need the vaccine if we develop the vaccine?
Oh, actually, HPV, the morphology of the HPV, when we do the PEP test, we diagnose, we can determine the protein that have in their uh, L, protein within their genome. So we can detect if the patient have a HPV 52 or not.
You can detect that thing earlier.
Earlier, no.
Okay, this is just the question to make sure you can think what is the function of vaccine actually. Go back and check what is the function of the vaccine. Okay, by having that specific vaccine, why we need to give the injection to the patient and so on. Tak apa, nanti balik bincang dengan your supervisor about that thing, about the
Okay.
concept of vaccine. Okay, that's all from me, Dr. Asmida.
Okay, thank you, Dr. Diana. And also thank you to Nurul Ain Farhana. Alright, our next presenter is Nur Aina Afika.
Can you see my slide, Doctor?
Yes, we can see your slide and your time starts now.
Assalamualaikum and a very good evening. I bid to all lecturers and my fellow friends. Okay, I would like to present my review paper entitled A Review on Biodegradation Study of Disposable Face Mask. My name is Nur Aina Afika binti Nur Azam. I've been supervised by Associate Prof. Dr. Siti Norasma Suri. This is my presentation outline. So today's discussion will cover all these parts. For the introduction, the 2019 novel coronavirus pandemic has been a huge problem in this world right now. So the regulation of wearing a face mask has been a must and have given rise to the pollution of the waste from face masks and microplastics. Thus, it cause a huge problem in the future. These are my research questions. First, how face masks will contribute to the environmental problems. Second, what are the factors that influence the degradation of the face mask? Third, what are the ways in reducing the waste from face masks? Fourth, what are the major findings in this review? And the fifth, what are the research recommendations that can be done in the future? Problem statement, 1.1 billion pieces per daily face masks used by the people in Eastern Asia, while 7.05 million pieces per daily face mask used by our Malaysian people in Southeastern Asia. Thus, it will lead to the abundance of the waste of face masks. This is a bar chart of Asia region's general population of face mask usage from data throughout 2020. This is a data of all regions in Asia, including Eastern Asia, Southern Asia, Southeastern Asia, Western Asia, and Central Asia. Covered the total daily face mask use in million pieces. So next significance of study is to review on face mask usage in Asia and to provide information on factors that contribute to the degradation of the face mask. Next, my objective of this study are to analyze the waste contributed by face masks and to study factors that help hasten the degradation of the face mask. First of all, what is face mask? It involves filtering the breath air and is made from a thin disposable material, functions to prevent contamination of patients and also function to prevent infection of healthcare personnel. Polypropylene is a thermoplastic polymer from a monomer of polypropylene. It has a 40 to 60% of crystallinity. The limited resistance to oxidizing agents, aromatic and halogenated hydrocarbons, and it also has a high heat of temperature distortion and stability. Next, the degradation of the face mask. Research conducted by Culturable et al. Said that the degradation of polypropylene-based face masks take place at the polypropylene unstructured region. In most of the cities that have a high-tech scientific landfills, unsanitary landfills, and also dumping yards. Landfilling is one of the ways to degrade this face mask naturally. So the result will be biofilm formation on polymer surface. Next, microbial community on face mask. Microorganisms such as Aspergillus niger, Pseudomonas and Vibrio species will attack the surface and form a colony of biofilm light. And then we do analysis on scanning electron microscope analysis. The results are 10 times larger abundance of microorganisms in soil has been observed on the surface of the sample. Next, factors influence the degradation of face masks. We have 4. First is the type of soils. We have garden soils, we have dumping subsoils. So these kind of soils have different rate of degradation. Next is PP amorphous region, which is it acts as a catalyst in photodecomposition process. Next is the microbial communities. We know that there are a lot of microbial communities in the soil, so this is one of the factors that will hasten the degradation. Next is the types of degradation. We have a lot of types, which are biological degradation, thermal degradation, mechanical irradiation, chemical, and so on. So these kind of degradation have a different rate of degradation of the face mask. Next ways in reducing the waste from face mask rotation, which we can hang the face mask to dry or keeping them in a clean, breathable container between uses. Next, recycling the face mask. We have 2 types, which is primary recycling and secondary recycling. Primary recycling is we reuse the face mask itself, while the secondary recycling is from the thermoplastic face mask. Next is the biodegradable face mask. We replace the polypropylene with organic and biodegradable materials such as bioplastic polymers. Next is the face mask filters of biodegradable plant fibers. Filters from plant fibers of polysaccharides such as sugar can also be used to change it with the woven technology of polypropylene face mask. There is also one type of filters which are from tea leaf waste filters. That can be done because it have a polypropylene in the tea leaf waste. Next, conclusion, the total face mask daily use in human population in Asian regions with the highest is in Asia with 1.11 billion pieces, followed by Southeastern Asia, Western Asia, and Central Asia. This scenario will cause severe pollution problems to human and environment. There are also few factors that affect the degradation, such as types of soils, microbial communities, polypropylene amorphous region, and the types of degradation. So my recommendation is to have the future study regarding the impact of face mask to the environment. Next, we can have a research to test the microbial effects on polypropylene-based face masks. We also can study the effects polypropylene face masks disposed in marine ecosystem other than terrestrial ecosystem. Next, to examine for other alternatives that can reduce the face mask waste. These are my references. It's all from me. Thank you.
Thank you, Aina. Do you have any question from our panel? Dr. Diana or Dr. Farul Ansfar? Kalau tak ada, saya nak tengok result, Aina. Okay, Dr. Diana ada. Okay, Dr. Diana.
Okay, Aina, show me your objective. Okay, you said that you want to analyze the waste contributed by the face mask. Okay, now what is the result that suitable for this?
Oh, I'm sorry, Doctor. The slides are before the objective itself. This bar chart is the result for my objective, for my first objective.
But this slide is not answering your objective. Betul tak, Dr. Diana?
Yeah.
Macam Dr. Diana ada.
Okay, sorry. It has some technical issue dengan my laptop and also my handphone ni. Okay,
tengok dia dua.
So, alright. Talking about this graph, I don't think this is answer your first objective.
Oh.
Is it?
Yes, I agree with you, Dr. Diana, because this slide is not answering the objective at all. There is no evidence to show that this is the waste that you are looking at caused by the usage of the mask. Betul tak, Aina? Sebab saya tak nampak from your results, any of your results which is answering any of your objective. You have two objectives and your results is not answering your objective at all. Dia macam more to you review the usage of face mask but not the waste caused by face mask.
Sorry, this is because it should be the waste usage, waste from face mask usage.
Okay.
Can I just comment kat situ? Sorry, Dr. Diana, you nak cakap apa?
Tak apa, you continue sekejap. I nak
I see. Okay, so I think the data they presented here is actually the mass usage. So the mass usage, but pieces in million. But the thing that you want to study is the waste. So how much waste from the usage? How much is the percentage of the waste from the usage? Okay.
Oh, okay.
So you need to know. Okay, if you can get the data. Let's say in Eastern Asia. Okay, for instance. Okay, it's about 1.1 to 5 million. Okay, pieces. But how much is the waste that is being generated by this 1.1 to 5 million face mask, okay, that have been used by the consumers? Okay, right. Okay, that's all from me. Okay.
Later I'll put the answer in my written review.
All right. Okay. Cool.
Have a look at your second objective. Okay, to study the factor that help fasten the degradation of the face mask. Show us the result for this one.
This is the only slide for this, but I have further explanation in the written report itself.
But this is showing the types of kejap the types of apa ni orang kata macam faktor yang menyumbang kepada degradation, right?
Yes.
Betul ke? Okay. But your objective is how to make the degradation go faster. The degradation process go faster. Betul tak? Objektif you macam tu.
Yes.
Right dan left.
So this should be the slide factors that influence by means to make the degradation faster. Should be influence the degradation of face mask and also give effects on the rate of degradation itself.
Yes, but if you look at the slide here, you have a jumbled up of everything. Like for example, you have types of degradation, and then kemudian tiba ada soil microbial communities. I know that soil microbial communities is a few of the factors that affect the degradation of the face mask. Tapi kat tepi ni ada type of degradation pula. Oh tak? Kat sini ada PP amorphous region. Like what you are trying to reflect in this slides, kita tak nampak sebab dia macam a mixture of everything. You nak cerita apa sebenarnya? You want to describe to us different ways, okay, to make the degradation process go faster, okay, or different types of a factor or different types of soil on which within the soil you have different types of microbial communities on which that alone will help to orang kata speed up the degradation process. Sekarang ni your review ni macam kelam kabut sikit lah. Okay, tak menjawab objektif tu. Okay, you have to sit down with the supervisor, look at the title, look at the objective, make sure it tallies, okay, with your results. Okay? Alright, thank you Aina. Our next presenter is Andrea Marcel. Okay, Andrea Marcel, anak Daniel.
Yes, I'm here.
Okay, you can start sharing your slides.
Okay, can you see my slide?
Okay, yes, you can see your slide and your time starts now.
Good afternoon to all lecturers and my fellow friends. My name is Andrea Marcel Anak Daniel, and today I will discuss a review on alpha-glucosidase inhibitory activity of plant species from family Polygonaceae for possible treatment of type 2 diabetes mellitus. Diabetes mellitus is a metabolic chronic disease that happens when the blood glucose level in the body is elevating abnormally. This condition is known as persistent postprandial hyperglycemia. There are two types of diabetes mellitus, and the main focus for this review is the type 2 diabetes mellitus that specifically caused due to the unresponsiveness of the body towards the insulin production. It is highly prevalent and can lead to other chronic diseases such as retinopathy, atherosclerosis, and neuropathy. According to the International Diabetes Federation, 463 million people suffered from type 2 diabetes mellitus in 2019. And it is assumed to increase up to 51% in the future. The research question that will be answered in this review are: what are the aims of the review? What is the gap of the study? What are the main findings from the review? What are the conclusions for this review? And what are the recommendations of this review? The aim of the review is to summarize the inhibitory activity of the alpha-glucosidase enzyme of 10 selected plant species from the Polygonaceae family, and to summarize the bioactive compounds that drive the anti-alpha-glucosidase properties from the plants. The other aims of the review is to determine the effect of different substrate and extracts of them in the alpha-glucosidase inhibitory assay. Acarbose, the drug used in the treatment of diabetes mellitus, have been causing few side effects such as flatulence, diarrhea, and abdominal discomfort. The efficacy of the acarbose is also declining for long-term use. Hence, the search for a new natural treatment for type 2 diabetes mellitus from plants, such as plant species from Polygonaceae family. These are the 10 plant species that have been chosen for this review due to its bioactive compounds that can contribute to medicinal use and can exhibit good alpha-glucosidase inhibitors. All of the alpha-glucosidase inhibitory activity of each selected plant species expressed with IC50 value or percentage inhibition together with their different parts of the plant use, extraction technique, and different extract solvent use has been simplified in the table shown in this slide. Please take note that a low IC50 value or high percentage inhibition when compared to the acarbose indicates strong inhibitory activity against the enzyme. For the second table, all of the bioactive compound extracted from the Polygonaceae family with their IC50 of alpha-glucosidase inhibitory activity has been summarized. Moving on to the analysis for each of the plant species, the first one is Polygonum multiflorum. Two different substrates are used, which are PNPG and maltose. And from the result, it shows that using maltose as a substrate, the plant extract inhibit the enzyme more effectively than PNPG. This is because maltose has higher sensitivity and specificity since the product released after hydrolyzing maltose is glucose. As for the bioactive compounds, imodine, which is an endocrinon, showed the strongest inhibitory activity against the enzyme. Next, Polygonum caspitatum. According to result, the plant root extract effectively inhibit the enzyme. This may be due to its bioactive compound extracted such as procyanidin B233O digallate and epicatechin gallate, which are classified as tannins and were found to exhibit strong inhibition against the enzyme. Moving forward, in the studies of Muehlenbachia temlifolia, Polygonum hercanicum, Rumex acetosella, Polygonum avicularis, and Rumex crispus, the results show that when using polar solvent, it gives better result compared to the other extract solvent in the alpha-glucosidase inhibitory assay. In terms of the bioactive compound, linoleic acid extracted from the Muehlenbachia temlifolia, and transcaffeoltyramine, calocatechin, and myrisatrin extracted from the Polygonum hercanicum, catechin extracted from the Rumex acetosella, myrisatrin, quercetin, and polydatin extracted from the Polygonum avicularis, and fiscian extracted from the Rumex crispus are the most potent compound among all the bioactive compounds extracted from each of the plant species. Next, Rumex hastatus, Phagopyrum esculentum, and Phagopyrum tetricum. These three plant species extract shows that it has higher IC50 value than the acarbose, which indicates that it has weak inhibition against the alpha-glucosidase enzyme. However, the bioactive compound extracted from the plant species show that all three plant species contain compound that possesses anti-alpha-glucosidase properties, such as flavonoid from the Rumex hastatus and quercetin for both Phagopyrum esculentum and Phagopyrum tetricum. In conclusion, all of the selected plant species from the Polygonaceae, except for Phagopyrum esculentum, Phagopyrum tetricum, and Rumex hastatus, showed significant inhibition against the alpha-glucosidase enzyme. And all of the selected plant species contain bioactive compounds that possesses anti-alpha-glucosidase properties and can become the potential alpha-glucosidase inhibitors. Lastly, the different substrate and extract solvent will affect the result of the inhibitory assay and may give false positive result. The limitation that I've encountered during this study is the lack of studies regarding the in vivo alpha-glucosidase inhibitory activity of the plant species from the Polygonaceae family, which only Polygonum multiflorum have the in vivo studies of alpha-glucosidase inhibitory, which was conducted several past years ago, which may get different result if conducted now with new and modern technologies that may improve the result. Next, the difficulty in extracting the data from some journal articles, as some journal articles only incorporate graph of inhibition without stating the exact value for IC50 or the percentage percentage inhibition. As for the recommendation, it is recommended to use polar solvent to obtain better result because it was proven that it gives better inhibition towards the alpha-glucosidase enzyme than the other extract solvent. Next, the use of maltose as substrate compared to the PNPG is better because it has higher sensitivity and specificity, which help to avoid false positive result. Lastly, more study on in vivo alpha-glucosidase inhibitory activity of the plant species from Polygonaceae family to determine the exact dosage for the new drug and treatment of type 2 diabetes mellitus. This is my reference, and that's all from me. Thank you for listening.
Okay, thank you, Andrea. Do we have any question from the panel?
Hi, Andrea. Can you go to the recombination slide just before?
The recombination slide?
Slide before.
Is it this one?
Yes. All right. Now, you keep mentioning that polar solvent give better result.
Yeah.
Now, I would like to ask you, which polar solvent that are you referring to? The polar solvent that extract the sample, or the polar solvent that we use to dissolve the extract?
The polar solvent that we extract the plants, the plant parts that we choose.
Okay, means that the polar solvent for the extraction.
Yes, extraction.
Okay. Can you give me some idea or suggest or discuss with us now why polar solvent give a better result?
Because as from the result,
you have mentioned about the compound and so on just now.
Oh, I see. Okay. When using polar solvent, the bioactive compounds that in the plant species are preserved because prior to the extraction, because when you use non-polar solvent, the bioactive compounds in the plant cannot really be extracted and the concentration may be different when the result using the polar solvent.
Does flavonoid is suitable for the polar?
As from the result, yes, because it used methanolic extract.
Yes, exactly. Okay. Show me your aim or objective of the review.
Okay.
Okay, stop. Wait. Okay, you are talking about IC50 and also percentage of inhibition.
Yes.
Okay, based on your review, okay, which is the best to be used in the reflect or in suggesting the inhibition activity? Which is the best?
Between IC50 and percentage inhibition?
Which is the best?
From my review, the best between that between the IC50 value and the percentage inhibition, it's better use the IC50 value because when using percentage inhibition, the dosage are not we need to know the dosage for the how can we inhibit the how many percent that we inhibit, we need to know the dosage for the inhibition process.
Do you need to run the for the percentage of inhibition? Do you need a single or multiple concentration, Andre?
You mean single or multiple?
Concentration means single or various concentration.
Single.
Single. So how about the IC50? But tadi you kata from the percentage of inhibition we can determine 1 point. Okay, how about IC50? So just to give you the idea, the hint, which is the best, then you can include the statement in your review. But memang we can see when we go through or read through the article, ada yang report in IC50 and then ada yang report most cases in percentage of inhibition. So that's the reason that you have to include which is the best, IC50 or percentage on revision. So next time kalau nak buat untuk alpha glucosides activity, they know, okay, they should report IC50 or percentage on revision.
I see.
Okay.
Okay.
Alright.
Thank
Go
you,
to the.
Dr. Diana.
Alright.
Thank you, Andrea. Alright, our last presenter for today is Siti Khatijah Rahmat.
Assalamualaikum, Doktor.
Salam.
Hello.
Okay, Khatijah, you may start sharing your slide.
Sekejap Saya takut start.
Okay, yeah, we can see it clearly. You may begin.
Sekejap Okay. Bismillahirrahmanirrahim. Assalamualaikum and a very good evening to all the panels and audiences. My name is Siti Khadijah binti Rahmat from AS201 6A3, and today I will be presenting my final year project, which is a mini article presentation with the introduction. The problem statement of this review would be the knowledge and information regarding food poisoning is available. However, the extent of such information is still scarce in developing countries. Malaysia is one of the developing countries and most foodborne cases are not reported, resulting in underestimated cases. The data of foodborne cases are unreliable and inaccurate, which gives rise to the ineffectiveness of public health priorities and food safety controls. Hence, the significance of this review would be to highlight the importance of having knowledge in food poisoning. The objective of this review, apart from the importance of knowledge, is to emphasize the seriousness of food poisoning in Malaysia. So the 5 research questions of this review would be: what is the significance of the study of food poisoning? What are the causes of food poisoning? How often does an outbreak occur in Malaysia? Where do food poisoning outbreaks usually happen? And what are the ways to reduce the number of cases in Malaysia? The gaps and limitations of this review would be gaps in years. Unreported cases across the country resulted in missing data. Apart from that, one of the gaps in knowledge is further research about infectious organisms. For example, the epidemiology of Shiga toxin producing Escherichia coli, staph and Salmonella in Malaysia. Before we delve deeper, a little bit of definition. Food poisoning is an illness caused by consuming foods that are contaminated, usually with viruses, bacteria, and parasites that produce toxins and can be fatal if left untreated. The common symptoms of food poisoning are stomach aches, cramps, diarrhea, vomiting, nausea, and fever. These symptoms can lead to even worse symptoms if untreated, such as diarrhea for more than 3 days, bloody diarrhea, and high fever. So now we move on to the main body of this review. Basically, this review is divided into 2 parts. The first part is determinants of food poisoning, which are infectious organisms, cross-contamination of food, and risk factors. So one of the determinants of food poisoning is infectious organisms, which are microorganisms that can cause diseases in any susceptible host, in which bacteria contribute the most to the spread of foodborne illnesses. The most common pathogens are Shiga toxin producing E. coli, staph, Salmonella species, Clostridium species, and Staphylococcus species. Viruses can also cause food poisoning, such as noroviruses. Apart from those 2, parasites such as Taenia solium, Toxoplasma gondii, Trichinella spiralis, and Echinococcus species are common parasites that can cause food poisoning. Another determinant of food poisoning is cross-contamination. This is the mode of transmission of infectious organisms. Cross-contamination is the accidental transfer of microbial agents from one substance to another that is often harmful and can happen during food preparation, food handling, processing, transportation of food, sales, and storage. In Malaysia, most cases resulted from or restaurants. For example, in 2016, there was an outbreak occurred in a residential school in Perak, which was caused by cross-contamination of pancakes from an external supplier. The pancakes were contaminated during its mixture process, along with short cooking time and low temperature for the pancakes to cook. Next, risk factors. Risk factors are defined as causes that increase susceptibility. The groups that are at risk of getting food poisoning are the elderly, children, immunocompromised people, and females that are pregnant and breastfeeding. Now we move on to the second part of this review, which is the frequency and distribution in Malaysia. The number of cases in Malaysia from 2015 to 2019 have increased from 15,300 and 346 cases to 17,147 cases, which Food Safety and Quality Report in 2019 showed that the incidence and mortality rates for food poisoning are the highest compared to other food and waterborne diseases such as hepatitis A, typhoid and paratyphi, cholera and dysentery. The incidence and mortality rates of food poisoning are 50.90 and 0.03 respectively. These are cases that are reported. However, there are many other cases that go unreported. One of the reasons is why a complex of chain called as population exposure occur prior before the reporting of a case. However, as of June 7th, 2021, the number of food poisoning cases in Malaysia have decreased with 44.2% throughout 2020 from 512 cases to 288 cases. Now we move on to the discussion of this review. From the review article, the determinants of food poisoning are identified, such as the role of infectious organisms, and the groups that are at risk in contracting food poisoning. The frequency and distribution in Malaysia can be seen increase throughout the years. However, the cases last year have decreased. Nevertheless, the accuracy of data is still lacking and actions must be taken to solve this problem. So how do we curb food poisoning outbreaks from happening? One of the suggestions is, in my opinion, is to revise statutes governing food safety regulations and management. The consequences of food poisoning can be lowered by intensive intervention, surveillance and monitoring from the government as well as private sectors. Next, awareness and knowledge should be spread to the public, especially rural areas where resources are limited. Public awareness should be made so that cases can be reported and monitored and preventive measures can be made. To summarize, the places that are at risk, that are often at risk in Malaysia are schools and restaurants, whereby inappropriate food handling procedures can be the main cause for handling. Sufficient knowledge about food handling among food handlers and good sanitation and hygiene to avoid the risk of contamination of food to be served to consumers. Foodborne illnesses are a burden, and it is in the hands of the government and the people as well to ensure the safety of food consumption and create a healthier environment free of foodborne diseases. These are the references that I used for my review article. And with that, I end my presentation. Thank you.
Thank you, Khadijah.
Welcome.
Okay, I would like to invite Dr. Diana or Dr. Kara Asfar to ask any question to ask a question to Khadijah.
Assalamualaikum, Khadijah.
Waalaikumsalam, Doctor.
Just my simple advice, not really the comment lah. The advice. Okay. Can you show me back your objective? The second slide, I think. The objective.
Kejap, Doktor tengah lag.
Tak apa, it's okay. Alright. Okay. Here you put just a very simple objective. Okay, you just to emphasize the seriousness of food poisoning in Malaysia. Okay, but in the content of your review, you also touch about the infection, microorganism, and so on, isn't it?
Yes,
Okay.
Doctor.
Alright. When you come to the conclusion, you detail out what you already reviewed hopefully in your written report. Okay, so I think you have to maybe rewrite, reconstruct your objective, and otherwise you have to reconstruct your conclusion, because the conclusion must answer the objective. But in this stage, in the current presentation, what I can see, your conclusion does not answering your objective. Okay, this is the main issue. Right. Show me the infectious organism. The slide that discussing about the infectious organism.
Kejap doktor.
Tak apa.
Tengah like.
Alright. You have this. You have bacteria, you have viruses, and also parasite. Okay. From this part, okay, can you tell me which is the most or the major culprit for your issue, food poisoning issue?
The major infectious organisms that contribute are bacteria.
Bacteria. So, have you discussed in your written report?
Yes,
so,
doktor.
sekarang you just mention macam ni je tau. So, we didn't know which is the most contributor risking factor. Itu.
Oh,
So, you
okay.
have to mention clearly in your written report since I'm the second examiner. So, I really hope, I expect to read your justification and also explanation regarding to this point. Alright, Rija?
Okay, doktor.
Okay. I think that's all
Thank
from
you,
me.
Doktor. Thank you, Dr. Diana. I do agree with Dr. Diana regarding your objective. Okay, you should you should have a new objective which tally to your results and also your conclusion. Okay?
Doctor, I
based
have
on
a question.
Yes.
Sila.
Kiranya my objective should be more specific ke apa?
Yes, more specific and then must align with your conclusion. Kalau tidak, you kena tulis balik conclusion.
Sebab if you look at your result, you have list out factor apa benda tadi, ada dua benda kan you mention kan. Satu types of organism causing food poisoning. Satu lagi sebab saya perasan ada dua yang you mention. So those could be your objective. And then bagi menjawab objektif tu is the result tu. And the conclusion tu based on result yang tadi you mention tu.
Okay.
Nampak tak? So ada dua lah yang saya perasan ada dua. But you have a good look at your slides, at your results and reconstruct your objective based on that.
Objective. Okay, doktor.
Alright.
Boleh.
Thank you so much, Khadijah. Thank you so much all the panels.
Thank you, Doctor.
You're welcome. Dr. Diana, thank you. Dr. Karasfa and all the students who has been with us since the beginning. Boleh tak saya nak minta semua orang on the camera so that we can take a picture sebagai bukti, and all of this recording, all of the presentation will be uploaded in YouTube channel. You will be getting a minimum of 3 marks lah based on this presentation. Kejap saya nak change layout kejap. Saya minta orang lain tolong ambilkan gambar juga just in case, sebabnya saya takut saya punya tak jadi ke apa ke. Sound. Okay, sebentar. Okay, everybody smile. Okay, 1, 2, 3. Okay, so thank you so much students. So I hope you will have a good rest. Dah habis dah kan semua kan? Dah habis dah untuk presentation for this one. Thank you so much and okay, satu lagi pesanan saya lah. Okay, just in case if you are having difficulty to finish up on time, do discuss with the supervisor. Okay, kalau rasa macam dah stress sangat ke, tak sempat sangat ke, do discuss with the supervisor, and you do know that UiTM has extended the deadline for a student to submit the assessment. Okay, yang kesian kat sini pensyarah tau, kita orang tak di-extend, so kita dipendekkan masa untuk menilai supaya you all will have more time. Kami pun sakit kepala, but that's fine. What we can do for now is that we want to make sure that you do undergo all of these processes as safely as possible, gain as much information as possible, tapi at the same time look after your health and your well-being. Okay? So kalau ada problem, bincang dengan supervisor. Kalau rasa supervisor tu garang sangat tak boleh nak bincang, cari je mana-mana lecturer yang you rasa can help you with that. Boleh? Tak dengar suara pun?
Boleh doktor.
Boleh doktor.
Okay. So with that, with that, I thank you. Kita tutup majlis kita dengan tasbih kifarah dan surah Wal-As. Okay, thank you so much. Thank you guys.
Okay, thank you guys.
Thank you, Doctor.
Thank you.
Best all everyone.
Assalamualaikum.
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 Okay, bye.